காக்கை குருவியை போல் கவலையின்றி நீ இருந்தால் யாக்கை கொடுத்தவனை யார் நினைப்பார் இவ்வுலகில். சட்டியிலே வேகின்ற சத்தெல்லாம் சரக்கானால் மட்டின்றி படித்து வந்த மருத்துவருக்கு வேலை என்ன. கடலருகே வீட்டிருந்தும் கடுந்தாகம் வரும்பொழுதே கடவுளெனும் ஒருவனது கைசிறக்கு நினைவு வரும். இன்னதுதான் இப்படிதான் என்பதெல்லாம் பொய்கணக்கு, இன்னதுதான் இப்படிதான் என்பதெல்லாம் பொய்கணக்கு, இறைவனிடம் உள்ளதடா எப்போதும் உன் வழக்கு. எல்லாம் அவன் செயலே என்பதற்கு என்ன பொருள், எல்லாம் அவன் செயலே என்பதற்கு என்ன பொருள், உன்னால் முடிந்ததெல்லாம் ஓரளவே என்று பொருள், உன்னால் முடிந்ததெல்லாம் ஓரளவே என்று பொருள் என்ற கண்ணதாசனின் வரிகளை துணை கழைத்து அந்த பிரபஞ்ச பேரியக்க சக்தியை வணங்கி தொடங்குகின்றேன். Good morning, ladies and gentlemen. I take this wonderful opportunity to render my deep sense of gratitude to the benevolent management of MGR College, my beloved principal, and the opportunity rendered by the Department of Biotechnology. Warm New Year greetings from MGR College, Osur. These decorated days would be meaningless if it is not occupied by wise men. To decorate the days further, ladies and gentlemen, put your hands together to invite our dignitaries to the days. First of all, I wish to have Dr. G. Needharajan, convener of this international conference, to the days. Next, I am very much delighted to call upon our beloved principal, Professor A. Muttumani sir, to the stage. I am pleased to invite our chief guest, Dr. P. Sundaresan sir, to the pulpit. I am very much elated and excited to invite our chief guest, Professor Govardhan Muthuswamy sir, to the dais. Thank you, sir. The beautiful spring came, and when the nature resumes her loveliness, the human soul is apt to revive also. To revive ourselves academically, we feel honored in offering you a cheerful welcome to the inaugural ceremony of DST-sponsored First International Conference on the Significance of Gain and Loss of Function and Impairing Transcription Factors Causing Genetic Disorders in Human. The way to bring about change is to be proactive and active, says Octavio Spencer. After organizing 8 funded national conferences in a row, Department of Biotechnology has decided to upgrade their organizing skills to the international level. Yes, ladies and gentlemen, today, the red letter day of the MGR College history, as it is going to witness its first international conference. We are what we repeatedly do. We are what we repeatedly do. Excellence, therefore, it is not an act, but a habit, opines Aristotle. Department of Biotechnology has the habit of knocking the doors of funding agencies regularly, and they have been successful all the time. Yes, ladies and gentlemen, International Conference on the Significance of Gain and Loss of Function and Impairing Transcription Factors Causing Genetic Disorders in Human is sponsored by DBT. At the outset, we believe that the knowledge sharing and worthy discussion in this conference and warmth in our hospitality would nullify the chillness of Osur at least for next couple of days. Have a sense of pride in your motherland, just as your mother has given to pay. Have a sense of pride in your motherland, just as to you, so as the land given birth to you, says Satya Sai Baba. To pay our respect to this wonderful motherland, Tamil Nadu, I kindly request you all to rise for the invocation, Tamil Thai Vaalthu. Go closer.
நீராரும் கடலுடுத்த நிலமடந்த கெழிலொழுகும் சீராரும் வதனமென திகழ்பரத கண்டமிதில் தெக்கணமும் அதில் சிறந்த திராவிடனல் திருநாடும் தக்க சிறு பிறை நுதலும் தரித்தனரும் திலகமுமே அத்திலக வாசனை போல் அனைத்துலகும் இன்பமுற எத்திசையும் புகழ் மணக்க இருந்த பெரும் தமிழனங்கே தமிழனங்கே உன் சீரிழமை திறம் வியந்து செயல் மறந்து வாழ்த்துதுமே வாழ்த்துதுமே வாழ்த்துதுமே.
Thank you. Settle down, please. Light symbolizes the auspiciousness, prosperity, and abundance. Another dimension of fire, one of the Panchabhutas, which has strong influence on the progress of any civilization. We witness this in the early part of the human life, when it began at the Stone Age. The progress of those Stone Age people only occurred when they discovered fire for the first time. Similarly, for the better progress of this first international conference, without any hiccups, I kindly invite all the dignitaries on the dais to inaugurate the international conference by lighting the Kuttuvilakku. Thank you, sirs. Thank you, ma'am. Pleasantries. Matru Devo Bhava, Pitru Devo Bhava, Acharya Devo Bhava, Aditi Devo Bhava, says our culture system. Aditi Devo Bhava, it means the guest is equivalent to the God. Honoring the guest always the hallmark of this culture system. We are happy and humble to recognize and honor the guest on behalf of our management and principal, sir. First of all, I kindly request our beloved principal, sir, to honor and greet our chief guest, Dr. P. Sundaresan, sir, senior scientist, molecular genetics, Aravind Medical Research Foundation, Madurai. Next, I benevolently request the convener of this conference, Dr. G. Neetharajan, to greet and honor our chief guest, Professor Govardhan Muthuswamy, sir, Professor by Research, Kyongpuk National University, South Korea. My invitation to Dr. K. Suresh to accolade our beloved principal, Professor Dr. A. Muthumani, sir. Thank you, sirs. A smile is the universal welcome, says Max Eastman. To have a formal welcome address, I would like to call upon an ever-smiling personality, mastermind of this first national conference, Dr. G. Nithirajan, Convener, Head Department of Biotechnology, to give away the presidential welcome address.
Thank you, Yuvraj. A warm and cheerful morning to everyone present here. It gives a great pleasure to start this auspicious occasion by heartily welcoming everyone for the DBT sponsored 2 days international conference on significance of gain and loss of function and impairing transcription factor causing genetic disorders in human. The theme of this 2-day international conference is genetic disorders caused by loss of gain of function and impairing transcription factor, which is important in today's world as there is a significant risk in getting the genetic disorders. To throw the light on these domains, we have invited eminent speakers to enlighten us with the field of specialization. My heartiest welcome to our eminent speakers, my PhD guide, Dr. P. Sundaresan, Dr. Govardhan, Professor J. Martin from Scotland, and Akshaya Murugesan from Finland, Arjun Masurkar from USA, and Ruby Vijaykumar from London, and Dr. Satish Kumar Rajshekharan from Israel, and Ganga Varu Subramaniam from Bangalore. I welcome all the speakers and guests from different colleges and universities all over the world, from different walks of the life, who will train by sharing their knowledge in the vast experience. We are happy to announce that the international conference is sponsored by Department of Biotechnology, Government of India. The main outcome of this conference will be cumulative knowledge gained in understanding the rare genetic disorders in their mode of function. We hope this two-day international conference would be help participants to expand their mental horizon, to enrich them with the knowledge from all over the world. I request all the participants to make use of these 2 days to the best of their abilities. With this brief note, I welcome our beloved principal of MGR College. I extend my chief guest, Dr. P. Sundaresan sir, Senior Scientist in Molecular Genetics, Aravind Medical Research Foundation, and Dr. Govardhan Muthusamy, Research Professor in Yangpu National University, South Korea. I submit my winsome welcome to the respected heads and faculties from various departments, distinguished researchers and participants all over the world, dear and colleagues, and my dear students and media persons. I welcome you all on the behalf of PG and Research Center in Biotechnology, MGR College, Hosur. A special welcome to Dr. Manivasagam, Head Engineering Biotechnology. Finally, I welcome all the enthusiastic participants around the globe for taking part of this conference. Thank you.
Thank you, sir, for your sweet, soothing, and scintillating welcome address. Release of souvenir and seeding. The abstracts of poster and oral presentation of the participants are recorded as souvenir, both in the form of CD and a hard copy. I wholeheartedly welcome the dignitaries on the days to release the same at this forum. I graciously welcome our chief guest, Dr. P. Sundaresan sir, to release the hard copy of souvenir of this conference. Then I kindly request Professor Govardhan Muthuswamy sir to unveil the soft copy of the souvenir CD. Thank you, sirs. This eminent personality believes that a dream does not become reality through magic. It takes sweat, determination, and hard work. The man who shed a lot of sweat with a determined heart. Our leader. I am proud and privileged to extend my warm invitation to our Principal Professor Dr. A. Muthumani sir to deliver the presidential address. Sir.
Very good morning to one and all present here. Most respected Chief Guest, Dr. Sundaresan sir, Dr. Govardhan Muthuswamy sir, HODs of various departments, faculty members and students, faculties from other colleges, students from other colleges. A warm welcome to each and everyone. By presenting here, you cannot sit here, and the prominent person who are sitting on the dais also cannot be sitting here. So the opportunity I got to stand before you. I don't know anything about that biotechnology, but I heard about a lot. In India, the infrastructure facility is not there. Like that, the chief guest, two persons they are telling. ஒரு 25 வருஷத்துக்கு முன்னால பயோடெக்னாலஜி டிபார்ட்மெண்ட் எங்கே இருக்குன்னு யாருக்குமே தெரியாது. இதில் என்ன பியூட்டி அப்படின்னா, டாக்டர் சுந்தரேசன் சார் இஸ் ஃப்ரம் மதுரை. ஆனால் ஹீ விசிட்டட் ஆல்மோஸ்ட் ஆல் த கண்ட்ரீஸ் இன் த வேர்ல்டு அண்ட் ஸ்ப்ரெட் த ஃப்ராக்ரன்ஸ் ஆஃப் தமிழ்நாடு பீப்புள். Another personality who is sitting there, Govardhan Muthuswamy. நான் நார்மலாக எந்த ஃபங்க்ஷனில் பார்த்தாலும் என் பேர் எதிலையாவது லிங்க் ஆயிரும். ஒன்றில் முத்து இருக்கும், அல்லாட்ட மணி இருக்கும். இன்றைக்கி ரொம்ப ஹாப்பி, முத்துசாமி நம்ம கூட இருக்கார். ஹீ இஸ் ஆல்சோ ஃப்ரம் தமிழ்நாடு, தட் டூ ஃப்ரம் சேலம். அவர் எங்கே ஒர்க் பண்ணார்னா சவுத்து கொரியாவில். நான் அவர்கிட்ட நக்கலாகவே 2 கேள்வி கேட்டேன். நீங்கள் எந்த கண்ட்ரின்னு? தமிழ் அவ்வளோ தெளிவாக பேசினார். சேலம்னாரு. இப்போ எங்கே இருக்கேன்னு? சவுத் கொரியா அப்படின்னாரு. சரி, எங்கேருந்து போனீங்க? நான் சவுத் கொரியா அப்படின்ட்டார். சார், தமிழ்நாட்டில் இருந்து போய்கிட்டு என்ன சார் இப்படி சொல்லுதீங்க? ஸோ யூ ஹவ் லேர்ன்ட் எவ்ரி திங் ஃப்ரம் ஹியர். ஸோ அவங்க தெளிவாக சொல்லிட்டாங்க. சுந்தரேசன் சார் தெளிவாக சொன்னார், ஐ ஹவ் ஸ்பெண்ட் எ லாட் ஆஃப் இயர்ஸ் இன் அதர் கண்ட்ரீஸ். ஐ வாண்ட் டு சாக்ரிஃபைஸ் த ரெஸ்ட் ஆஃப் த லைஃப் டு தமிழ்நாடு. அண்ட் ஹீ இஸ் ஒர்க்கிங் இன் அரவிந்த் ஐ ஹாஸ்பிட்டல், மதுரை. We cannot get the people like this, but we, including you, got the opportunity. நான் சார்ட்ட சொன்னேன், மார்க்கோனி ரேடியோவை கண்டுபிடிச்சார் அப்படின்னு எத்தனை பேருக்கு தெரியும்னு கேட்டால் 10% சொல்லிடுவீங்க. சொல்ல மாட்டேன்லாம் சொல்ல மாட்டேன், சொல்லிடுவீங்க. ஆனால் அந்த ரேடியோவை எத்தனை பேர் பார்த்துருக்கேன்னு கேட்டால் ஒருத்தர் கூட பார்த்துருக்க மாட்டேன். ஆனால் இப்போவும் ரீசெண்டாக அந்த ரேடியோ இந்த ஃபேஸ்புக்கு யூடியூப்லலாம் அது வந்துக்கிட்டு இருக்குது. அப்பப்போ அதை காணிப்பாங்க. ஸோ நாங்கள்லாம் சிட்டி வில்லேஜில் இருக்கச்சில் ஒரு ரேடியோ மட்டும் வச்சுருப்பாங்க. எல்லாம் அக்ரி ஃபார்முக்கு போயிட்டு சாயந்தரம் 6 மணிக்கு மேலே சாப்பிட்டுட்டு 7 மணிக்கு மேலே அங்கே பஞ்சாயத்தில் ஒரு ரூம் கட்டி கொடுத்துருப்பாங்க. அதில் உட்காந்து கேட்டுக்கிட்டு இருப்போம். கேட்கறதுல பாதி புரியாது, ஏன்னா அவன் பேசுறது பூரா ஹிந்தி. நம்மளுக்கு தமிழே பாதி புரியாது, இங்கிலீஷு அதை விட புரியாது. அப்போ நம்மளுக்கு என்னதான் தெரியும் அப்படின்னா, மொபைல் மட்டும் தெரியும், அதில் படம் பார்க்க மட்டும் தெரியும், ஆனால் அதோடைய வேர்ட்ஸை நான் படிக்க மாட்டேன். ஸோ ஃபேஸ்புக் இஸ் தேர், YouTube is there. We never got that opportunity to went through it, but you are getting the opportunity and you are not utilizing it. That's what sir was told me. Why the students are like that only? they are younger. Their age is like that. Age will come and go, but the time you never will take charge of your life. And I should thank each and everyone. கொஞ்சம் ஹார்டாக தான் இருக்கும், லைஃபே ஹார்டு தான். எல்லாம் எப்பவுமே சந்தோஷமா இருக்க முடியாது. கஷ்டம் வந்தால் தான் சந்தோஷம் தெரியும். எப்பவுமே சந்தோஷமாக வாழ்ந்தோன்னு வச்சுக்கா கஷ்டம் தெரியாது. ஸோ லைஃப்பில் அப்ஸ் அண்ட் டவுன்ஸ் இருக்கும், சாரோ இருக்கும், ஹாப்பினஸ் இருக்கும், ரெண்டும் இல்லாமலும் இருக்கும். ஸோ யூ ஷுட் ஸ்ட்ரைக் அவுட் எ பேலன்ஸ் பிட்வீன் ups and down and sads and happiness. I have great pleasure in extending greetings and good wishes for the organizer for the successful completion of this international conference on genetic disorder in human. Most of the human beings are always in dishonor and unitic, but this is a topic like that, how you are going to make it as and change it as a human being. So come up in your life as a good human being. Thank you.
Exactly, sir. You have to compare with others sometimes. That is why the great poet Kannadasan said, உனக்கும் கீழே உள்ளவர் கோடி நினைத்து பார்த்து நிம்மதி நாடு. Thank you very much, sir, for your presidential address. Next, I take an immense pleasure to invite this stalwart in this arena, who contributed his research life to bring enough consciousness and awareness not only among researchers, but also in the public of this country. An eminent scientist, an able administrator, a multifaceted, iconic figure of the field of molecular genetics, mentor of my mentor, our chief guest, Dr. P. Sundaresan sir, for his inaugural address. Sir.
Thank you. So, very good morning. See, if I stand over there, you can't able to see me. That's what I'm saying. I'm standing in the center, okay? So very good morning. So first of all, I would like to say my sincere thanks to the organizing committee members, especially the MGR Medical College, all the nucleus members of the MGR Medical College, especially the principal. I had a nice interaction with this morning briefly, but he is very sweet and we had a nice interaction. And then obviously I have to say my special thanks to my first PhD student, Dr. Nidhi Rajan. And other faculty members. So I think here there are so far 25 PhDs awarded from under my guidance. There are 2 students are here. One is Dr. Nidhi Rajan, another one is Dr. Nalathambi. So he is my first PhD student and then he is my third PhD student. Okay, so with this introduction, I am giving a motivation because I don't have much time. So the other speakers should be interact with you. So what I'm going to do, I'm going to briefly interact with the 5 to 10 minutes. That's it. Okay. So I think you already studied the biology, molecular biology and everything, because you are studying the biotechnology. So I will talk in Tamil later. So concerned with like a molecular biology, you should know what is central dogma. Central dogma is nothing but the DNA is the genetic material. From the DNA is coming to the messenger RNA. From the messenger RNA it is coming to the protein. Okay, if that the process is perfect, you are normal. Okay, so everyone here, I think you are young, energetic and then you are healthy. So that it means your gene, okay, your gene is, there is no mutation. So that's what you are without any disease. That's what here principal told us, disorders. You don't have any disorders. Okay, once is there any mutation in the DNA, obviously it won't give the right protein, so it will form a abnormal protein that lead to the disease. So once the disease may occur in the early part of life or the child born with the defect, okay, or if you get aged 35, 40, 50, 60, then the disease will come. Okay, so now to understand, to avoid the disease, because we have to live healthily and then people are living more than 100 years now. If you go to Japan, in the Tokyo itself, more than 200 persons, they are living more than 100 years. That is called centurion. Centurion project. Okay, so you take the DNA and then find out what kind of gene is responsible for the longevity of the life. So like that, you have to understand your own DNA. Your own DNA. Once you understand, you can lead your life healthily. So that is the message I want to take, point number 1. The second one is I'm going to give you some 4 advices. A little progress each day adds up to big results. Okay, so you no need to study the end of the semester or the end of the test. Suppose the test is on Monday, you don't worry about all the 5 days. You are going to study either Saturday or Sunday. Don't do that. Okay, when the teacher started to teach, you take the keynotes. That's what you are doing in the Google. I think, Thambi, why are you laughing? I want to interact with you after the talk, okay? Otherwise you go out of this class or the room, please. Okay, a little progress each day adds up to big results. That's what I'm telling. You no need to study end of the semester, and that whatever the teacher is teaching on the class, you better to have it in mind, and then you take a kind of keywords. That's what you are putting in the Google. You put the right Google word, and then you can pull down so many information. So that's what I'm telling. Better to acquire knowledge in a small way, and then that definitely it is going to make a big difference. A little progress each day adds up to big results. That is point number 2. And then the success is not straight line. Success is not straight line. Okay, I don't know how many of your parents are illiterate. They're not yet graduated. I don't know. If it's so, can you raise your hand? My father is not graduated. I am the first graduate. I think you have to say, you have to be very proud. You have to be very proud. Okay, so that you have to pay more. Gratitude to your parents. So like that, I think that is to show that your gratitude, what you're supposed to do, you have to come up in life in a big way. So I'm also belongs to a poor family, and the no education, we studied in Tamil medium, I am standing here. Okay, so the success, again I'm telling, success is not a straight line. Okay, simply if you have money, even you can't, if you have money, you can't able to succeed. It is very difficult to succeed. Okay, so what I'm saying is, lot of difficulties will be there. You overcome all the difficulties, and then you reach your goal. That's what I'm telling. Success is not a straight line. Work hard, and then you achieve your goal. Okay, so that is point number 3. And then another thing is, I think I'm going to give you 2 options. If you want to be a unique personality, either in the classroom or in the society or in your working environment, if it's so, you have to be very, very unique. அப்பனா தனித்தன்மை, உனக்குள்ள ஒரு தனித்தன்மை இருந்தாதான் நீ மிளிர்வ. எல்லா ஸ்டாரா இருக்கலாம். போலார் ஸ்டார் இஸ் டிஃபரெண்ட். All are stars. Polar star is the unique. Okay, so if you want to be a polar star, okay, so you have to little bit different. Okay, so option, I will give you 2 examples. One is like Ozur. This is Ozur, right? இதை வேற மாதிரி திங்க் பண்ண முடியுமா? இப்போ முனீத்ராஜன் சொன்னார் என்கிட்ட ஜஸ்ட் 2 days ago. ஐ திங்க் நான் உங்ககிட்ட முன்னாடி பேச போகிறேன். பட் என்னுடைய சிந்தனையை தட்டி விடணும். I have to turn on my somebody in the I think he talked about Kannadasan. Kannadasan, he will come to enter into some marriage hall. They will give some topic on the spot. He will write the all anything. கரித்துணி, தொடச்சி போட்ட கரித்துணி இருக்குல்ல, அதை பற்றி ஒரு கவிதை இருக்கு. பியூட்டிஃபுல் கவிதை. ப்ளீஸ் ரீட் டுநைட். ஸோ அது மாதிரி ஒசூர் யாராவது இஃப் யூ ஆர் திங்க் அவுட் சைட் த பாக்ஸ், யாராவது சொல்ல முடியுமா? அப்படி உங்களுக்கு மனசுக்குள்ள வந்ததுன்னா யூ இன்ட்ராக்ட் வித் மீ திஸ் ஈவினிங் ஆர் டுமாரோ, வில் டிஸ்கஸ். ஓகே, அது பாயிண்ட் நம்பர் 1. ரெண்டாவது கல்லூரி. திஸ் இஸ் அ காலேஜ். நோ, கேன் யூ ஏபிள் டு திங்க் அபவுட் கல்லூரி? College, you have to think differently. Can you able to do it? Please, if you have spark within you, உனக்குள்ள ஒரு ஃபயர் இருக்குன்னு வச்சுங்க, அது வேணால் என்கிட்ட எழுதி காட்டுங்க, நான் கொஞ்சம் அதை பற்றி படிக்க போகிறேன் அப்புறம். ஓகே, ஸோ த ஃபோர்த் பாயிண்ட் இஸ் யூ ஹாவ் டு பி, இஃப் யூ வாண்ட் டு பி யூனிக், பெட்டர் டு பி திங்க் அவுட் சைட் த பாக்ஸ். ஓகே ரைட். இப்போ வந்து ஒசூர் நான் எப்படி திங்க் பண்ணேன்னா HOUSUR, தட் இஸ் த ஸ்பெல்லிங், கரெக்டா இல்லையா? HOSUR, ஒசூர். The pronunciation of ஒசூர் is maybe different. That is called artificial intelligence. Now you no need to type anything in the laptop. When you speak to the computer, any word it will recognize, and then it will tell you, yes, this is ஒசூர். Okay, ஒசூர். They will tell, am I right or wrong? One guy from Korea, he can't able to pronounce correctly the ஒசூர். Okay, so like that, what I thought, நான் உங்களுக்கு இது வரைக்கும் நிறைய அட்வைஸ் பண்ணியிருக்கேன், அந்த ஒசூரே அப்படியே மாற்றி போடலாம்ல. ஓ ஷோர் இல்லையா? நான் இது வரைக்கும் நான் வந்து உங்களுக்கு அட்வைஸ் பண்ணியிருக்கேன், அந்த ஸ்பெல்லிங்கை மாற்றுங்க. HOSUR. ஓகே, இந்த ஓவை எடுத்து முன்னாடி போகிறீங்க. ஓகே, OH அதை அப்படியே பிரிக்கிறீங்க. SURE, E வந்து சைலண்ட், அதனால ஓ ஷோர். This is Dr. Sundaresan. Okay, you have to think outside the box. That's what I got the award from President of India. Okay, if we have spark within you, please tell me end of the day or tomorrow. Okay, I think I don't want to take much time. I am a very good poet. I written so many poems. So Needharajan told, why not you speak in Tamil, because my students love Tamil. So why not you speak in Tamil? I am going to read about a poem, whatever I written about college. In your point of view, please tell me later, we will discuss. College, இதோட முடிச்சிடறேன். காலேஜ், அது ஒவ்வொரு சென்டென்ஸையும் ஒவ்வொரு இதையும் நான் 2 தடவை படிக்க போகிறேன். கல்லூரி, த டைட்டில் இஸ் கல்லூரி. பள்ளியின் அண்ணன், திங்க் பண்ணியிருக்கீங்களா? பள்ளியின் அண்ணன் கல்லூரி, பிகாஸ் யூ ஆர் ஸ்டடிங் இன் தொடக்கப்பள்ளி. நீ வந்து உன் தம்பி வந்து ஸ்கூலில் படிக்கிறான், நீ வந்து காலேஜில் படிக்கிற. ஸோ பள்ளியின் அண்ணன் கல்லூரி. நான் வைரமத்தோடு அதிகமாக திங்க் பண்ணணும், என்னைய நானே தட்டி கொடுத்துருக்கேன் நிறைய தடவை. ஐந்து ஆண்டுகள் விளையாடும் இளமை மைதானம். யாருக்காவது புரிஞ்சுதா? 5 ஆண்டுகள் விளையாடும் இளமை மைதானம். 3 வருஷம் பிஎஸ்சி, 2 வருஷம் எம்எஸ்சி. நீங்களாம் ரொம்ப ரொம்ப டீனேஜில் இருக்கீங்க. 5 ஆண்டுகள் விளையாடும் இளமை மைதானம். ஜாலியாக இருக்கீங்களா? என்னங்க, எஸ் சார் நோ? எஸ். நெக்ஸ்ட், தேர்ட் பாயிண்ட், தன்னைத்தானே செதுக்கிக் கொள்ளும் சிற்பக்கூடம். தன்னைத்தானே செதுக்கிக் கொள்ளும் சிற்பக்கூடம் அப்படின்னா, டீச்சருக்கு வந்து இருக்காங்கன்னு வச்சுங்க, அவங்க டீச் பண்ண மாட்டாங்க. யூ ஹாவ் டு டீச் யுவர் செல்ஃப். இஃப் தே கிவன் த சிலபஸ், தட்ஸ் இட், தேர் ஜாப் இஸ் ஓவர். யூ ஹாவ் டு எஜுகேட் யுவர் செல்ஃப். ஸோ தன்னைத்தானே செதுக்கிக் கொள்ளும் சிற்பக்கூடம். ஓகே, ஃபோர்த் பாயிண்ட், நிஜ சிற்பங்கள் சிரிக்கும் இடம். இங்கே கேட்ச் இருக்கு. நிஜ சிற்பங்கள் சிரிக்கும் சித்திரக்கூடம் அப்படின்னு யாருக்கா தெரியுமா? நிஜ சிற்பங்கள் சிரிக்கும் சித்திரக்கூடம். விவேக் பாணியில் சொல்லணும்னா, புரிஞ்சிக்க, புரியாதவன் புரிஞ்சவங்க கேட்ட தெரிஞ்சுக்க. அப்படின்னா என்ன தெரியுமா அர்த்தம்? நான் என்ன நினச்சி எழுதுனா, பொண்ணுங்களும் அழகாக இருக்காங்க. நிஜ சிற்பங்கள் சிரிக்கும் சித்திரக்கூடம், இது வந்து கோஎட்களுக்கு வந்து பொருந்தும், பாய்ஸ் தனியாக இருந்தால் பொருந்தாது. புரிஞ்சுதா இல்லையாங்க? என்ன யாரும் ரியாக்ஷனுக்கு தரப்படுங்க, இந்த பக்கமாக ரியாக்ஷன் வரணும். நிஜ சிற்பங்கள் சிரிக்கும் சித்திரக்கூடம். அடுத்த பாயிண்ட் வந்து, ஆசான் இருந்தும் ஆசிரியர் ஆகும் இடம். யூ நோ நீட் டு டிபெண்ட் ஆன் டீச்சர்ஸ். யூ எஜுகேட் யுவர் செல்ஃப் and then you form a good group. You read one chapter, another person read another chapter, and then you discuss together. That time you are becoming a teacher by yourself. So ஆசான் இருந்தும் ஆசிரியர் ஆகும் இடம். சிலருக்கு மலைக்கு ஒதுங்கும் மண்டபம். படிக்காத பசங்க இருக்கானுங்களே, அதுக்கு தான் இது. சிலருக்கு மலைக்கு ஒதுங்கும் மண்டபம், பலருக்கு அடுத்த பாயிண்ட் பாருங்க, பலருக்கு கனவை நனவாக்கும் தெய்வம். இந்த கை தூக்குனாங்கல்ல, உங்க அப்பா எவ்வளவு கஷ்டப்பட்டு உங்களுக்கு அனுப்பிச்சிருக்காங்க. ஸோ யூ சப்போஸ் டு ப்ரூவ் தை ட்ரீம், ரைட்? ஸோ பலருக்கு கனவை நனவாக்கும் தெய்வம். அப்புறம் அடுத்து ஃப்ரெண்ட்ஸ், நண்பேண்டா. ஓகே, என் நண்பன் கிடைத்த தோட்டம் இல்லையா? இங்கே கிடைக்கிற ஃப்ரெண்ட்ஷிப் வந்து இட் நெவர் எண்ட், இட் கீப் கோயிங், கீப் கோயிங், கீப் ஆன் கோயிங். ஓகே, என் தோழன் கிடைத்த தோட்டம், உயிர் நண்பனை காட்டிய நன்னீளம். காலேஜ். ஓகே, கலைமகளுக்கு விடை கொடுத்து, அடுத்து வந்து கேம்பஸ் இன்டர்வியூ வராங்கல்ல, அதெல்லாம் கொண்டு வந்துட்டேன். கலைமகளுக்கு விடை கொடுத்து மகாலட்சுமியை வீட்டுக்கு வரவழைத்த இடம். கைதடுங்க. கலைமகளுக்கு விடை கொடுத்து மகாலட்சுமியை வீட்டுக்கு வரவழைத்த இடம். அடுத்து இங்கே மகன் பட்டமளிப்பு விழாவுக்கு கைரேகை தந்தை வந்த இடம். சாதி மத பேதமின்றி சங்கமித்த இடம், புவி சரித்திரத்தை மாற்றும் இடம் இந்த கல்லூரி. தேங்க்யூ.
The pain of a placement officer lies in the question every interview process. What is your USP? Unique selling point. Exactly, sir. Thank you very much for the wonderful poem and your intellectual, inspiring and immaculate inaugural address. Thank you. Next, I would like to invite this a passionate professor, panoramic scientist, the chief guest of this inaugural ceremony, Professor Govardhan Muthusamy, sir, professor by research, Kyongpook National University, South Korea, for his keynote address. Sir.
அனைவருக்கும் இனிய காலை வணக்கம். So good morning ladies and gentlemen. Today we are gathering a special occasion. So DBT sponsored international conference on the significance of gain, loss of function and impairing transcription factors causing genetic disorders in human. So first of all, I congratulate the organizing team for organizing this wonderful international conference, so which will covers genetic disorders in human health. So as well as you are young budding students, you will know, you can take some points about your health. So what are all the some of the environmental factors, so which will I am going to give in my lecture. So what are the impact of some environmental factors to cause genetic disorders and also human health. So I hope you will enjoy this two-day international conference with various experts in this field of biotechnology as well as medicine. So once again, I congratulate the Department of Biotechnology, MGR College, Hosur. So I thank for inviting me as a resource person for this wonderful occasion. So I hope I will give some points to take with you. So see you in lecture. Thank you.
Thank you, sir. He is looking very young, right? But he is having more than 4 or 5 postdoctoral fellowship in various universities. Thank you very much, sir. Gratitude is the essence of a quality life because the humans are always dependent. A timely expressed thanking will always bring happiness. In turn, a happy heart always radiate a positive energy that will bring the divinity to your doorsteps. To record our gratefulness, I would like to invite Dr. J.M. Murugan to propose the vote of thanks.
Thank you, Professor. எனது காலை வணக்கத்தை தெரிவித்துக் கொள்கிறேன் அனைவருக்கும். Good morning, one and all present here. Always have an attitude of gratitude, says staring ground. I, Dr. J.M. Murugan, standing before you to propose the vote of thanks at this urgent gathering. At the outset, I am immensely thankful to our respected chief guests of the inaugural ceremony of this first international conference ICSFGD 2023. Dr. P. Sundaresan sir, and Dr. Govardhan Muthusamy sir, welcome, for having accepted our invitation and gracing these occasions. Thank you, sirs. A special mention to our respected principal sir, Dr. A. Muthumoni, for being the catalyst that stimulated us to do our best. Thank you, sir. I would also like to thank the convener and the organizing committee of the conference who worked behind the scenes to make this event successful. Our sincere gratitude goes to all the HODs, staff members of our institutions for their rock solid support and encouragement. My behalf of thanks are always due to all the dignitaries of our and neighboring institutions whose presence made this event much productive. Finally, I submit my sincere acknowledgement to our student volunteers, placement officers, media persons who made this event a memorable one. Once again, I thank you to all.
Thank you, sir. தனம் தரும் கல்வி தரும் ஒரு நாளும் தளர்வரியா மனம் தரும் தெய்வம் வடிவம் தரும் நெஞ்சில் வஞ்சமில்லா இனம் தரும் நல்லன எல்லாம் தரும் அன்பர் என்பவர்க்கே கணம் தரும் பூங்குழலால் அபிராமி கடைக்கண்களே என்ற அபிராமி பட்டரின் வரிகளை துணை கழைத்து வாய்ப்புக்கு நன்றி பாராட்டி விடைபெறுகிறேன். So this concludes the inaugural ceremony of this first international conference, followed by all the technical sessions. I kindly request all the delegates to be seated, and the technical sessions will be handed over to Dr. Kirtana.
A very good morning to everyone present here. And to our online participants too, a very good morning. To have a feast to our eyes and ears, now we would like to call upon Priyadarshini, first BCom CA, for a classical Bharatanatyam dance. Please put up your hands to invite her to the dais.
வரவேற்போம் வண்ண வண்ண கோலம் தீட்டி வரவேற்போம் வசந்த கால மலர்கள் தூவி வரவேற்போம் வண்ண வண்ண கோலம் தீட்டி வரவேற்போம் வசந்த கால மலர்கள் தூவி வரவேற்போம் வண்ண வண்ண கோலம் தீட்டி வரவேற்போம் வாசல் வந்த வான்மழையாய் வரவேற்போம் வாசல் வந்த வான்மழையாய் வரவேற்போம் வாசமிகு சொல்லெடுத்து வரவேற்போம் வாசமிகு சொல்லெடுத்து வரவேற்போம் வருக வருகவே வருக வருகவே ஆன்றோரே சான்றோரே வருகவே வருக வருகவே வருக வருகவே பெற்றோரே பெரியோரே வருகவே வருக வருகவே வருக வருகவே ஆன்றோரே சான்றோரே வருகவே வருக வருகவே வருக வருகவே பெற்றோரே பெரியோரே வருகவே வசந்த கால மலர்கள் தூவி வரவேற்போம் வண்ண வண்ண கோலம் தீட்டி வரவேற்போம் இதய மலர் இதழ் விரிக்க இன்னிசையில் பண்ணிசைக்க இரு கரங்கள் குவிந்திருக்க பதமலர்கள் நடுபுரிய சிரிக்க இன்னிசையில் பண்ணிசைக்க இரு கரங்கள் குவிந்திருக்க பதமலர்கள் நடம் புரிய சந்தன திலகமிட்டு ஆரத்தி எடுத்து வரவேற்க வந்தோம் சபைதனிலே பமபதா பமபதா பதா பதம பதமரிக சகரிகரி சரி சமாம சரி சபா பபா பதனி நிகரி சனி தபம பதனி சந்தன திலகமிட்டு ஆரத்தி எடுத்து வரவேற்க வந்தோம் சபைதனிலே கவி நிலவாய் கலைமகளாய் யாழிசைத்தோம் வருக கலை தென்றல் மனம் வீச மலர் சொரிந்தோம் வருக அரும்போர்க்க உறவாட களிப்புடனே வருக கண்ண சுவை கண்டிடவே உவப்புடனே வருக.
Very good morning. Very good morning to one and all present here. So thank you Dr. Kirtana for giving this opportunity to introduce the extraordinary and eminent professor Dr. Govardhan Muthuswamy. Am I audible? Am I audible? Okay, fine. So Dr. Govardhan Muthuswamy is a distinguished scientist and teacher who has made significant contributions in the field of environmental biotechnology. He is currently working as a professor in research, Department of Environmental Engineering, Coimbatore National University, Daegu, South Korea. He has completed PhD in Chikungpuk National University, South Korea. Dr. Govardhan Muthuswamy has over 18 years of research experience as well as teaching in the field of environmental biotechnology. He started his career as a postdoctoral fellow in the Division of Biotechnology, Chonbuk National University, South Korea, and in 2015, and he later moved to Department of Applied Science, Mureren Institute of Technology, Japan, and also he worked as a PDF fellow in Department of Environmental Engineering, University of Seoul, South Korea. To his credit, he has so far published more than 200 research articles in internationally peer-reviewed journals. Total impact factor of 1,121 impact factor. It's a such huge impact factor this science will contributed. Even the paper is cited for more than 5,000 citations. Really is the one of the member, like he got an award from that enlisted as a top 2% scientist in the world, published by Stanford University, USA. Currently 2021 to 22, he is enlightened that the worldwide 2% of the scientists, he is the one of the renowned scientists for with us today. And he has acted as a managing guest editor in several special issues edited by Elsewhere, Willy, and as well as Science Director journals. One of his research paper is featured in the Elsewhere top list journal, the hottest article in the Middle East Asia. And also he is serving as editorial in chief members from different journals like Science Director journals. He also awarded a research fellow sponsored by the National Research Foundation, South Korea. He received that many awards in that organization. Almost he got funded around 2 crores projects in that, particularly in that institute. So such kind of eminent professor with us. He has visited many countries as a researcher in the field of environmental biotechnology, and he is also acted as a resource person from that various conferences in all over in world, and especially last few months for the last few conferences like conducted by Manonmaniam Sundarar University and as well as Alagappa University and then Reva University from Bangalore. So he is busy man with us today. He gave that lectures to us. So he has vast experience in that research. With these short introductions, I am happy to invite Dr. Govardhan Muthuswamy to deliver his the talk from the title of genetic disorders in humans. Thank you so much.
ஸோ எல்லோரும் எப்படி இருக்கீங்க? ஸோ கொரோனாக்கு அப்புறம் வந்து வணக்கம் சொல்கிறதெல்லாம் முடிஞ்சிச்சு. எல்லோரும் எப்படி இருக்கீங்க? நல்லா இருக்கீங்களா? சாப்பிட்டீங்களா? எப்படி போயிட்டுருக்கு ஸ்டடீஸ்லாம் எப்படி போயிட்டுருக்கு? என்ஜாய் பண்ணுறீங்களா? ஸோ இன்றைக்கி கான்ஃபரன்ஸ் வந்து இன்டர்நேஷ்னல் கான்ஃபரன்ஸ் ஜெனட்டிக் டிசார்டர்ஸ் சம் ஹியூமன் ஹெல்த்து. ஸோ பேசிக்கலி I'm an environmental microbiologist. Link how the environmental pollutants will affect the human health. இல்லை தூங்கிட்டு இருக்கீங்களே? ஸோ wake up. ஸோ இது வந்து apart from research, it is an social awareness topic. அதாவது ரிசர்ச் வந்து ஒரு சைடு வச்சிடலாம், ஓகேவா? ஸோ ரிசர்ச்சுக்கு முன்னாடி இட் இஸ் a social awareness topic. ஸோ இதுக்கு வந்து ரிசர்ச் பண்ணி தான் நம்ம வந்து இதை சால்வ் பண்ணணுன்னு அவசியம் இல்லை. ஸோ இது அப்படின்னா அந்த டாபிக் என்னென்னா பிளாஸ்டிக்ஸ். ஸோ பிளாஸ்டிக்ஸ், மைக்ரோ பிளாஸ்டிக்ஸ். ஸோ உங்களுக்கெல்லாம் நல்லா தெரியும். ஸோ டே டு டே லைஃப்பில் வி ஆர் யூஸிங் தீஸ் டைப் ஆஃப் பிளாஸ்டிக்ஸ். ஸோ காலையில் எந்திரிச்சதுலேருந்து நைட்டு தூங்க போகிற வரைக்கும். ஸோ ஐ வாண்ட் கேரி பேக். ஸோ ஈவன் யூ ஆர் கோயிங் டு பர்ச்சேஸ் எ சிங்கிள் டீ பேக்கெட் ஆர் சுகர். ஸோ suddenly you are asking to the shop owner, I want carry bag. So I didn't bring any bag. So I want plastic carry bag. So do you know what are all the impacts of these kind of plastic bags in human health as well as environment? புரியுதா உங்களுக்கு? ஒரு இந்த பிளாஸ்டிக்னால புரியாது, சொல்கிறேன் நான். என்ன ஒரு விளைவுகள் வந்து நம்ம சமுதாயத்தில் ஒரு சுற்றுப்புற சூழலில் என்ன தாக்கம் ஏற்படுத்துது? தமிழ் ஓகேவா, இங்கிலீஷ் வேணுமா? எந்த மொழி உங்களுக்கு வேணும், தமிழ் ஆர் இங்கிலீஷ்? ஓகேவா? ஏன்னா நானும் தமிழ் மீடியம் தான். ஸோ ஐ நோ யுவர் ஃபீலிங்ஸ். ஸோ ஐ வில் எக்ஸ்பிளைன் இன் தமிழ் as well as இங்கிலீஷ். வீடியோ
What can we do? Recycling? Of all the plastic ever generated as of 2015, only 9% was recycled. Even when it is recycled, plastic degrades. Then a floor mat, even in the best case, it doesn't recycle, it downcycles. And then it becomes pollution that ruins our beaches and chokes sea animals forever. We should not be forced to pollute the ocean every time we eat, drink, or go shopping. We need to be given a plastic-free choice. This is not hard to imagine. Indeed, some responsible companies are already leading the way. There are throwaway plastic-free aisles in supermarkets. Beverage companies already offer plastic-free soda and salsa water. Restaurants offer plastic-free service. There are plastic-free rooms in hotels. An airline is now offering a plastic-free flight. And Oceania and our allies are winning plastic-free victories. Following campaigning by Oceania, Peru passed a law that keeps plastic out of national parks and beaches. Belize is getting rid of styrofoam. And the European Union has mandated reductions for throwaway plastic throughout its 28 member countries. But with your help, we can do more. We can reduce throwaway plastic. We can make sure we're all given real plastic-free choices. We can help our cities, towns, schools, and workplaces establish zones that are free of throwaway plastic. Let's stop plastic pollution from wrecking our beaches and our oceans, and have healthy oceans that delight and sustain us for generations to come.
So types of microplastics, primary microplastics and secondary microplastics. Smaller size in and are produced external human use. Secondary microplastics, they occur as a result of breakdown of large plastic debris. Next one. So you can see the effect of microplastics in water and aquatic system. So எப்படி plastics வந்து enter ஆகுது food chain அப்படின்னு பார்த்தீங்கன்னா, same we are throwing the plastics by the action of UV and mechanical microbial. So these injected microplastics will enter into the seafoods. So obviously we are getting seafood, the microplastic transformation will occur in this way. Next one. So testing for microplastics in bottled water. இனிமே கொஞ்சம் இன்ட்ரெஸ்டிங்காக இருக்குது, அதை மட்டும் பாருங்கள், ஒரு 4 ஸ்லைடு. ஸோ நீங்கள் டே டு டே லைஃப் we are using many aquafina, Bisleri or Amma water, many waters. In that water itself, microplastics. You cannot find plastics in visual, eye visual. So, but in those water, researchers identified waterல தண்ணிலையும் microplastics இருக்கு. ஸோ என்ன பண்ணலாம்? நெக்ஸ்ட் ஒன் ஃபில்டர் பண்ணலாம். ஸோ நெக்ஸ்ட் microplastics in human health. ஸோ பின்னாடி எல்லாம் தெரியுதா உங்களுக்கு ஸ்லைடு? தெரியல தானே? எனக்கு தான் கண்ணில் பிரச்சனை. ஸோ hidden finance in the air. So microplastics, you are likely taking tiny particles of plastics every time. So you eat, drink and breathe according to the research in the risks of human health. So even nowadays in the air காற்று வந்து தூய்மையானதில்லை. Air itself containing some microplastic, nanoplastic particles. So while breathing, we are taking inside the microplastics. So next. So plastic pollution via soil, air and water. So these are all the main routes for entering the humans. So these plastics will affect headache, dizziness, diarrhea and liver disinfection, sometimes infertility, hormonal changes and cancer. Next one. So microplastics in human health, how இந்த ஏர்ல இருந்து எப்படி வந்து அந்த microplastics வந்து human bodyல enter ஆகுது. So what are all the disinfections in human health? Next one. So this is the important. So microplastics in child, from mother to child transformation. So this is very serious issue. So உலகத்திலேயே பாதுகாப்பானது தாயின் கருவறை சொல்லியிருக்காங்கல்ல, it's a safe place வந்து. But nowadays the microplastic transformation will occur from mother to child through placenta. So it is very shocking news. So recent reports are identified this phenomenon. So microplastics, the mother will take microplastics in any form from the toothpaste or water or some other food items. So will after mother through placenta, the nutrient will pass to the child, as well as the microplastic, nanoplastics also will pass through the placenta. So next one. So microplastics in breast milk, this is also very serious. You can maybe you read the news 1 year ago or 6 months ago. So microplastics in human breast milk. In the world, the purest form is breast milk. உலகத்திலேயே தூய்மையானது சிறந்தது தாய்ப்பால் அப்படின்னு சொல்லிட்டு இருந்தோம். But no more. So no more, it is not pure. So the scientists discovered human milk, breast milk also contain nanoparticles. So it is very it is quite challenging. Why I am saying this? As an environmental microbiologist, I am telling this microplastics entering human health through various forms, and we want to be safe. So we want to control the plastic pollutions. That's why I am telling the why, how microplastics are plastics affecting the human health. So next one. So microplastics found in blood. So blood. So overall, our total body is containing some microplastics, either blood or milk or placenta to mother to child through placenta. So we need to think about it. So it is just a news. For you, it is just a news. For you think about, already we are getting more diseases, genetic disorders and seasonal diseases. But this is burden. We want to think about this kind of pollutants entering into the body. So I am not a medical scientist, just I want to show you the transformation of this kind of pollutants in our body. So this is an awareness. So you should think about how we can stop this kind of pollutants entering into the body. So next one. So what is the solutions? ஓகே இவ்வளோலாம் சொல்கிறீங்க, பிளாஸ்டிக்ஸ் அங்கே இருக்குது, பிளட்டில் இருக்குது, சாப்பாட்டில் இருக்குது, எல்லாரும் இருக்குது அப்படிங்கிறீங்க. சரி, என்ன தான் பண்ணலாம்? வாட் இஸ் த சொல்யூஷன்? ஸோ ஹவு கேன் வி ஓவர் கம் திஸ் பிளாஸ்டிக்ஸ், ஹவு கேன் வி மிட்டிகேட் திஸ் பிளாஸ்டிக்ஸ்? எப்படி வந்து இதை ஒழிக்கிறது அப்படின்னு பார்த்தீங்கன்னா, ஸோ biological degradation. So as I mentioned earlier, physical and chemical methods is not applicable. அதாவது ஒரு பொருளை அழிக்கணும்னா முழுசாக அழிக்கணும், அதுதான் வந்து ஒரு சிறந்த தீர்வு. That is the successful rate of degradation. But in this case, you cannot completely degrade, you cannot completely eliminate plastics. எரிச்சாலும் வந்து இது, if you burn, it will release poisonous chemicals, so it will spoil the environment. And if you use some plastic, some chemicals to degrade, it is not possible. You cannot completely degrade. So that's why some eco-friendly method, we are using some microorganisms, either bacteria or some other microorganisms, the plastics. It is an eco-friendly method without any secondary pollution. So we are using biodegradation, nothing but degradation of plastics using some microorganisms. So next one. So these compounds are broken down into simple and natural compounds. So microplastic, most of the plastics are not biodegradable. So that's why we want to use some bacteria or fungi to biodegrade the plastics. Next one. So biological degradation is generally considered as a phenomenon green source, because in biodegradation using microorganisms there is no secondary pollution. So that is simple method. We don't want to spend much chemicals, we don't want to spend much labors. It is quite simple, eco-friendly and inexpensive. Next one. So you can see the overview of plastic degradation process. So polymer is nothing but plastics. So depolymerases, these enzymes are secreted by microorganisms. So these enzymes will cut the polymer into oligomers, dimers and monomers. So either aerobic or anaerobic process, this plastic degradation will occur. Next one. So totally 4 processes in biodegradation. 4 processes are involved by degradation of plastics. One is biodeterioration, biofragmentation, assimilation and mineralization. So this is the general phenomena how the microorganisms will degrade the plastics. So we can see one by one. Next one. So what is biodegradation? So biodeterioration is defined by undesirable changes. Did you notice that? So if you throw away some low density, carry bag, if you throw away these plastics in the environment, just watch it. After 6 months or 7 months, the structure will be collapsed, either mechanical or UV radiation. So on the structure பார்த்தீங்கன்னு வச்சுக்கோங்க, so it will tear. ஈஸியாக வந்து பிச்சிட்டு வரும், மண்ணில் இருக்கும்போது ஸ்ட்ரக்சர் வந்து கொலாப்ஸ் ஆகிருக்கும். So that is we can call biodeterioration. So next one. So assimilation characterize the larger fractions of molecules transported into the cytoplasm. So also mineralization refers complete degradation of molecules that will result into the excretion of oxidized metabolites. You can see that images. First picture பார்த்தீங்கன்னா polythene, அந்த low density polyethylene. So polythene போயிட்டோம்னா அதில் நிறையா இருக்குது. This one is low density polyethylene. Carry bag இருக்குல்ல அது, that one is control. Next one பார்த்தீங்கன்னா ஒரு 7 days or 15 daysல we are applying some bacteria. So we are treating some bacteria. So we can change the undesirable changes on the surface of the polythene, that polythene. So after 20 days, so we can see some major changes for the degradation rate of polythene. Next one. So you can see the microbial degradation of plastic process. So involved enzyme secretions. So maximum these microorganisms involved biodegradation is mediated by enzymes secreted by the microorganisms. Next one. So what is the significance of enzyme biodegradation? So which is eco-friendly. So again we want more quantity of enzymes. We want to scale up industrial microbiology. So once we find potential microorganisms which will have capacity to degrade the plastic, so we will get successful biodegradation of plastics. So you just think about the plastic pollution, so which is very serious problem in the world. So this is general topic and a social awareness topic. Wherever I can say it is social awareness topic. So just we want to alert our society. So please stop plastic, use some cloth bags. So we want to take responsibilities as a biotechnology, as a microbiologist. So we want to have some responsibility. So finally, if we are accumulating the environment, so the people or world will call us. We need microbiologists, we need biotechnologists. We want some solutions how to overcome or how to mitigate this kind of plastic pollutions. So finally, please avoid plastics, use some health bags, use some cloth bags. So thank you. Next slide. So apart from this, I want to share some points. புரியுதா இந்த இங்கிலீஷு படிக்கிறது அப்படின்னு பார்த்தீங்கன்னா you just gain the practical knowledge. So if you want anybody to get higher education in abroad, how to approach, how to get admissions, what are all the procedures, where can I ask if you want to apply some masters or PhD. So there are lot of fundings available in and around the world. So if you want any higher education doubts, so you can reach me at my email ID. So you can collect my email ID from your head of the department or Dr. Suresh Kumar. So I'm always happy to answer, to clarify your doubts regarding subject as well as your career. Thank you.
Thank you, sir. Thank you so much. Dear students, if you'd like to ask few questions, the session is now open for discussion. If you don't have any, I have one question. So recently we read in some papers that there are no microbes which are degrading plastic, sir. So how is that happening? Is it really feasible? So recently, students, please sit down. Recently, we have read in papers that there are microbes which are degrading plastic, sir. So how far it is going or how far it is applicable? How near are we in decomposing plastics using microbes, sir?
Hello.
So it is infinite. We cannot predict time, how long it will take to find some microorganisms. So it is not, as I mentioned, is quite challenging. So because of this density polymers, even the microbial systems are so collapsing. So we want to do some strain improvement. So then only it will be applicable. So we are trying to isolate some good microorganisms which will have potential to completely degrade. But so far, there is no report in the world.
How does the microbe use this plastic as an energy source or
energy source? Energy source. That is the basic. So any pollutants, the microorganisms will take as a nutrient source.
But that is it's completely polythene. How is it breaking down?
So due to the depolymerase action of some enzymes, the microorganisms will secrete.
And what is the monomer that it is taking inside for the energy, sir?
Monomers, oligomers and monomers take. So
the plastic is broken down into small monomers and it is consumed.
Oligomers, monomers.
Okay, sir. Okay. Thank you, sir. Students, do you have any questions? Okay, here we have one question.
Sir, whether it will completely degrade the plastic, sir?
It is not possible to completely degrade. It is not possible. There is no report. It is quite challenging. So even if you observe 10%.
If you don't have any questions, we shall wind up this session. And sir is available with us. You can ask your questions through email also. Thank you. Thank you, sir. Thank you. It was a very interesting and informative talk. Thank you so much for sharing your knowledge with us. And I know you are in a very busy schedule. Thank you for accepting our invitation too. Thank you once again from our side, sir. Thank you. Now it is time to move on to the technical session 2. So for starting this technical session 2, we cordially invite Dr. Sundaresan, sir. Welcome, sir. And to chair the session, I'd like to call upon Mrs. N. Soumya ma'am to please chair the session. Please take over the session, ma'am. Thank you.
Good morning to one and all present here. It's my pleasure to welcome Dr. P. Sundaresan sir and introduce to the gathering Sundaresan sir and introduce to the gathering here. He completed his PhD microbiology in Madurai Kamaraj University and also he served as a part doctoral fellowship in University of Pennsylvania, Georgetown University and assistant professor in School of Medicine in New York. He trained area research focus as molecular genetics related to eye diseases, diabetics and retinopathy, ocular albinism and a congenital retinopathy and releases cataracts. He also published more than 140 scientific research articles in various national and international journals. Sir is also instrumental for scholars as 24 research scholars. Sir also received Christ Award in 2010 and 11. And he has received 2 ICMR Award for Biomedical Research and Scientist Award. And Tamil Nadu Scientist Award.
Grandmother or uncle, they crossed more than 60 or 65, definitely they have some age-related diseases. Age-related diseases, the reason because their signature is signature in this sense, the DNA got mutated and then that particular thing, is coming to the next generation or it is late onset. Understand? That's what people are suffering. For that, next slide. Okay, so now I put nobody, in the previous talk, nobody asked any question. So that's what I'm telling, take your pad and pencil, whatever you thinking in your mind related to this particular talk or concerned with your research career, what I'm going to do, what kind of path I supposed to take, something like that. Now you write it in the notebook, you ask right now or we will discuss during the lunch time. Okay, so better to ask a question. Without asking question, I think, see, if they brought up like in US, the baby, they never suppress the baby. She is supposed to ask why I have to do it, what, all the question, they will, even it's a kind of culture from the childhood. But here the environment here in Indian scenario is different. If you ask a question, they're suppressing their individuality. So don't do that. So concerned with this particular topic, please ask a meaningful question. If it is a stupid question, I'm not going to answer anything. Ask a meaningful question related to this particular topic. You may have the problem or your family having some problem. Sir, this is the problem. This is a genetic effect. What is the cause? Can we able to stop it or is there any treatment? You can ask a question like that. Okay, so it means if you understand your own genome, if you know your entire genome, you can lead your life healthily till like a 70 or 80. Now the family, the family size is very, very small. Okay, so even when you get older, you can't able to depend on others. Nobody going to help you. Even nobody going to care your babies. That kind of situation is there. Okay, so that's what I'm telling. You ask a meaningful question. Okay, next slide. See here, very simple, like in the first grade, in the first grade, ஒண்ணாம் வகுப்புல படத்தை மட்டும் தான் போட்டு பாடம் சொல்லி கொடுப்பாங்க. The same thing, I am putting a simple diagram. See, this is normal, and this is mutated, right? One is normal bug, another one is mutated. It means my gene is mutated, that's what I am looking different from others. Okay, so where exactly the mutation is happening? This is the DNA, this is the DNA, exactly this is the original. If the gene is not mutated, it looks like this. If the gene is mutated, this is the correct copy and this is the mutant copy. If the mutant copy, this is the mutant copy. Okay, so you can compare the same thing to the disease, genetic disease. Okay, next slide. Next slide. Okay, so now this is the current trend is human genome and then personalized medicine. Human genome and then personalized medicine. In the sense, you take simply a drop of blood, one drop of blood from you, okay? We can isolate the DNA of your own DNA and then we can screen the entire genome of you, okay? Anybody knows how many genes we have, the human genes? Some approximate number. So we have, previously if you see the literature, 50,000 genes, 25,000 genes like that. Now that is not the scenario. The number of genes we have, 19,000 genes. 19,000 genes only in our body. So the 19,000 genes, it is doing various functions. So I am going to talk about only related to some of the genes. Okay, so here, as I told you, give a one, I think the madam is here. She extensively worked for this particular human genome project, and then she can able to, if you give the blood, she can able to screen, and then she will tell you what kind of mutation you have and what kind of disease you will prone at the age of 50 or 60. How to prevent or postpone the onset of the disease. You can't able to prevent. Somebody asked a question, can you able to remove all the bacteria? One faculty asked, concerned with the previous talk. It is impossible to remove all the bacteria. See, a good observer is a good scientist. You see advertisement in the TV, like a Colgate, that you are brushing the teeth. It is eliminate all the bacteria, and then if you see carefully, one bacteria sitting on the gum, even in the advertisement. It means it is impossible to remove the organisms. Okay, it means that is sticking in the gum. The same thing here, once you understand your genome, what you can do, yes, the DNA is mutated, I may prone to develop the disease at the age of 50 or 55 because my father had the same problem. Okay, so like that, how to postpone the onset. So that's what I discussed with the Nalathambi. Like concern with your father, mother and grandfather, definitely they were affected by diabetes. Don't take carbohydrate, avoid carbohydrate. Take less carbohydrate, more protein, more fat. Okay, so that will help you to reduce the sugar level in your blood. Okay, if you have that kind of education, see, I think those 4 people, they don't like my, I think 5, they are not liking my lecture, or volunteers, I don't know. Okay, so the personalized medicine, so that is the personalized medicine. I think anybody walked out, you can go out right now. Right? So once you understand your genome, you can postpone the onset of the disease. Consider the disease is going to appear at the age of 50. So if you follow that, yes, my gene is mutated, I am following some factors, you can postpone the onset of the disease at the age of 60 or 70. That in Bangalore, I think Nalathambi, that boss, he used a book, how to control your sugar. He is having a 300 rupees book, what to eat, what should not eat. If it's so, consider my age. My age is 75 or 60, something like that, but I am not 70, okay? So I am still mentally young, I am like a sweet 16, that is different story. So, you think the age is 70, but metabolically, the person is 60. It means 10 years he is younger than the actual age. So you have to make that kind of lifestyle. If it's so, once you understand your genome, then you change the lifestyle that will take, live longer, happily with your life. Okay, that is the personalized medicine. Even the cancer, okay, cancer, you know, some of the drugs may suitable for one particular person, some of them are not. Even the coronavirus, you know, it is mutating frequently. Some people are susceptible for corona, some people are not. But that is based on the immunity. If the immunity is very less, obviously that they will die. Okay, there are so many things, you know. Again, once you understand your personalized genome, that will helpful for the personalized medicine. Very recently in America, they, you know, the human genome project is completed long back, 25 years ago. Now they made another step. It means in 2030, within the year 2030, what we supposed to do, what we supposed to do to live longer or to live healthily. So what they did, you know, I told you 19,000 genes. That is only in the coding region. In the non-coding region, we don't understand anything. Okay, we have to explore the non-coding regions of our genome. If you understand the coding and non-coding region, you can even, you know, you can understand the pathogenesis of the disease like that, you know. Then everything is the clear cut. You know, you can understand the disease pathogenesis, and then you can go for a proper treatment. Okay, that is the human genome and personalized medicine. Next slide. Next slide. Okay, please recall the 10th grade and the 12th grade book. Obviously you got higher mark in biology. This is the structure of the eye. Okay, because I'm not going to talk about any other organ, because I'm working in eye hospital. I supposed to talk about the eye. So this is an eye. So this is cornea, this is lens, this is the anterior chamber, and this is the posterior chamber. This is retina, this is optic nerve. This optic nerve is going to the brain. Okay, so once you see the object, the light is coming through the pupil, it is in the macula, and then it is transforming the information to the brain. Yes, this is the bouquet, this is the water bottle. I can able to recognize the object. Is there any problem? Okay, so is there any problem in the cornea? It will mutate here and there. Obviously, some people lose the central vision, some people lose the peripheral vision. That is depending upon the disease. Okay, please, maybe you can interact with your parents, you can interact with your grandparents. They may have the glaucoma. Glaucoma is nothing but the eye pressure. Not only the pressure is in the heart, you know, there is a pressure in the eye too. Once the pressure is very high, it will affect the optic nerve. So the optic nerve here, the optic nerve is gone. If it's so, they will lose the peripheral vision. They can't able to see this pillar and this pillar. They can able to see only the center. So that is one disease. If there is any problem in the retina, if there is any problem in the retina, okay, so it will affect the central vision. They can see the periphery. They can able to see the center. Everything is so, you know, is depending upon the gene or mutation. Okay, so I am going to interact with you 3 diseases, all the work done by Nidhirajan and Nalathambi. Okay, that is their PhD topic. I am going to briefly explain in a detailed manner. Next slide. Okay, see here. This is I will come one by one. The disease number 1, 2, and 3. So this is number 1. So I think you see the eyelid. The eyelid is normal. If you see some of the patient, the eyelid is like this, is a very narrow down. See here, see the difference. This lady having a proper eyelid. This is the sister, her sister. See the eyelid. This is her son. This is normal, and again another one son for her, you know, the eyelid is like this. So it is depending upon a particular gene. Okay, so out of the 90,000 genes, one particular gene is defective for that particular in the family. So that is inheriting to the family. Okay, so here, okay, this is the normal one, sister abnormal, and her son is normal, and another son is having the defect. So like, you know, this is a genetic effect. Okay, this is number 1 disease. Another one, as I told you, glaucoma. So this is the structure of the eye. See here. So this is the iris and ciliary body. The ciliary body secretes the aqueous humor, a kind of fluid, that the fluid should go in the anterior chamber and then this is in a kind of open angle. It will go through, and then it go through the meshwork, and then epistellar vein. That is the pathway. So you and me having a proper open angle, the angle is open, so that's what the aqueous humor is flowing in and out. So based on this flow, there is no change in the pressure of the eye. If there is any blockage in this place, you know what it will do, one particular gene is normal, it will go. If it is abnormal, the gene is mutated, you know, it will form the protein interaction, it will form together, and then it will form a complex that is nothing but extracellular matrix, a kind of clump. So it will block, then the peripheral vision is gone. Okay, that is number 2. The third one, this is Nalathambi thesis, and this is Nidhirajan thesis. If you type simply aniridia, even now, simply type aniridia, A-N-I-R-I-D-A. If you type that one, this is, I am talking about, he did PhD in 2000. This is the first picture in the world. This is his picture. Okay, so in the Wikipedia, I think this is the reference for aniridia. So we reported long back, even some of the gene we identified. In the first time in the world, we identified some of the candidate gene for eye disease. We did it. So we published in the very high impact journal that is called Nature Genetics. Okay, so I will tell you those stories later. So this is PBES, and then this is glaucoma and aniridia. I am going to discuss in detail all the 3 diseases one by one. Next slide. Okay, so this is the first one. I told you we have the eyebrow is good, but some people having this kind of narrow eyebrow. You know why I am putting this one? This particular patient is from Huzur. They collected, huge family, 5 generation family, grandfather, father, mother, like that. 5 generation. If it is in US, they will migrate here and there. They can't able to collect the samples. But in India, they are not migrating, they are sitting in one place for 5 generation. Or you collect all the samples, and then you can do a excellent study. Okay, so like that, so we did all the screening. Then the name of the gene is called, if you write down FOXL2, FOXL2, that is the name of the gene. Okay, so the gene name is FOXL2. Okay, so see here, this is the structure of this particular gene. So this symposium or conference is concerned with the transcription factor related conference. So this gene is a kind of transcription factor gene. Okay, so here, and this is the gene structure, I think. Okay, so like recall the structure of the gene, the promoter, and then the coding regions, like that, and then it is having the ATGC start codon, and start codon, and the stop codon is a TAA. Please, you interact with your faculty, and they may take the class related to gene and gene structure. So this is what is exon, intron. This is the structure of the gene, promoter, and then transcript region. Next slide. Okay, so this is the how many number of. I am going to focus only 3 transcription factor genes. Please, go to next slide. Next slide. If you please note down this particular paper. Maybe I am requesting the faculties. You can give the assignment to all the students, and if they want to know, I think please go through this particular paper. It will give you all the information about the transcription factor. This is one of the excellent review. It will tell you the complete story. So what is transcription factor of the human? Okay, it will tell you all the stories. Go to the previous slide. So that, next slide. See, all the information will be there. So how many what are the families? Because I don't have much time. So what are the families? What is the number? All the information will be there. Anyhow, I am picked up 3. I am going to explain only 3 of the transcription factors. Next slide. Next slide. Okay. So now the first disease related to the eyelid, that is called that is called blepharomatous Tobis epicanthus syndrome. It's a very complex name. Simply write it down. PBES. This is that's what I told you is a keywords. Okay. So bodybuilder syndrome and then FOXL2. This is the name of the gene. So what we did, the functional studies were demonstrated the FOXL1 gene for PBES and reported first evidence for recessive bodybuilder syndrome. Recessive in the sense, I think you please recall what is autosomal dominant, what is autosomal recessive, what is X-linked recessive, and then mitochondrial disorder. Some disease, you know, if you see the five generation from grandfather to grandson, the disease, it is vertical transmission. In in all the generation, they have one member is affected in all the 5 generation, at least one. If it is a recessive case, grandfather is affected, his son is not affected, but grandson is affected. It means the recessive disease skip one generation, but the severity of the disease is very high for recessive. Okay, so one is dominant and then recessive, and another one is X-linked recessive, and another one is mitochondrial disorder. The mitochondria, you know, whatever the mitochondria, the ATP produced in our body, that is derived from mother. Whatever the mitochondria we are having, we got it from our mother, not from father at all. Some few repeats are there, that is different story. Mainly, whatever the mitochondria we have, we got it from mother. If the mitochondria is having some mutations, you know, definitely the son or daughter, definitely they have the disease. Okay, right? So here, okay, that is the recessive. I am explaining what is dominant, recessive, and then mitochondrial disorder. Next slide. Okay, so now we will see the FOXL2 gene. What is the FOXL2? What is the beauty of the gene? See here, FOXL2. This is considered a gene. So if you see gene, the amino acid sequences. Okay, protein is nothing but yes, amino acids. So here, in the particular gene, a kind of alanine repeats. See here, you and me, if you sequence the protein sequence or the DNA sequence, there are 17 to 18 alanine repeats. That is a very interesting thing. Okay, see here, you and me having the 18 alanine repeats. See here, this is polyalanine repeat, you know, it is keep on going. Understand? Alanine. But you and me have the same 18 alanine repeats in our genome, but those that narrow down, you know, that the diseased person, if there is any one more alanine is added, or one more or two more alanine is added, is a problem. Understand? A kind of, this is a FOXL2, it is having a alanine domain, polyalanine domain. Obviously, the norm, even in the normal gene, 17 alanine are there. If there is any problem in the particular patient, if there is one more alanine or two more alanine added, definitely no, they will end with the disease. That is the message. You got it? Yes? Next slide. Okay, see here, I told you this is all done by Nalathambi. This is published in a well impact journal that is called in human genetics, human mutation, like that. The comment, you know, suppose it is very difficult to publish the paper, but once you get accepted, the reviewer, you know, they will comment, you know, they will give very good comments. If from India, you know, it is going, the paper is going to abroad for evaluation, they never appreciate this particular paper, you know, what they told you, that is called biological relevance. Whatever you are doing in the bench, that should be translated through animals and then prove. The same thing here, the comment is, it is impossible to repeat in the animal model. Even this kind of experiment, you know, it is impossible to repeat in the animal model, because all the Americans, you know, or Europeans, they are doing all the experiments in the mouse. If you go to Switzerland, if you go to other places, you know, one mouse, they will put a statue for the mouse, and then the mouse, you know, it is having some DNA on the hand. You know, because, you know, they are giving that much importance to the mouse, okay, because sacrificing all the mouse for the biological experiments. But here, in the human, you know, it is impossible to get this kind of family. Okay, this is a kind of, excellent information we are giving to the scientific community. Okay, so this is the 5 generation family. See here, this is the husband, this is the husband. Please note this pedigree. I think again I'm telling the faculties, ask them to draw the pedigree, how to draw the pedigree, that kind of thing. So here, see here, this is the father, I think a person married elder daughter, sorry, the elder sister and then younger sister. He's a lucky guy. Yes? No. Are you with me? புரியுதா இல்லையா? ஒருத்தர் வந்து அக்காவையும் கல்யாணம் பண்ணிட்டாரு, தங்கச்சியும் கல்யாணம் பண்ணிட்டாரு. புரியலையா? நான் இங்கிலீஷ் பேசுறது புரியுதா புரியலையா? நான் தான் கத்தி எனக்கு நானே பேசிட்டு இருக்கேனா? So this person is married the elder sister and the younger sister. The elder sister having 3 sons. The second son is affected. He is the second son. His first son is normal. She is the normal mother. But the sister, the sister, she is having some maternal issue. Even she can't able to become pregnant because of the defect. She is infertile. Okay, we proved because of the mutation, because of the FOXL2 gene mutation, she is impotent, and then she can't able to have the baby. Okay, so here I think this boy having this kind of same variation like her. Okay, that is because the gene is sometimes it is expressed, sometimes it may not expressed. So the same way here in this family, this kind of phenotype is appeared. So I think we proved in a perfect way in everything like sequencing, gene expression studies, everything we did it. So this is the point number 1. Understand? That is FOXL2. Next slide. Next slide. Okay, so now we will come to the second disease, the glaucoma. Glaucoma, that is it is secreting the aqueous humor, and then the aqueous humor is going through the meshwork. If there is any blockage, they will lose the peripheral vision. I told you, right? So the gene name is called SIX6. This is primary open angle glaucoma. The primary open angle glaucoma, the name of the gene is called SIX6. Again, this is the transcription factor gene. Please understand, I am picking only the transcription factor genes for this conference. So here, I think this is the name, this is the gene, and we identified the mutation in different regions of the gene. Next slide. And see here, in the promoter region, not in the coding region, this is the promoter region. Promoter region is very, very important, where the transcription factor binds. Okay, so here, see here, these are the transcription factors, and we identified a 4 base pair deletion. In the previous one, there is 1 more or 2 more alanine added, but here deleted. It means a DNA, sometimes it may add, sometimes it may be delete. So here I given an example, addition in the previous one, here it is a deletion. So there is a 4 base pair deletion. Because of this, there is a defect. Next slide. Next slide. So you can prove in a zebrafish model. Whatever you are doing in the experiment, you have to prove that is called a biological relevance. You can prove it in the zebrafish model. So this is the way to do it. So you can take the SIX gene and then you can delete the 4 base pair, make a plasmid, and then put it in the vector. You take that one and then put in the zebrafish egg. I think I am going to ask you one question. The fertilization, what is fertilization? For fish, it is a external fertilization or internal fertilization? External, correct? So the male and female, and then the female it will release the egg, and then the male release the sperm. The fertilization occurs in the water, right? So you can take the egg, the fresh egg, and then you take that plasmid, with containing that the 4 base pair deletion, and then you inject that DNA into the egg. Okay, then you put it and then fuse together after the fertilization. Once the sperm is entered, that is fertilized within a short span of time. Next slide. So why we have selected that zebrafish? The zebrafish is very, very transparent. You can see in the naked eye. even in the globe, because we are working on the eye, the globe size, the globe, in the entire eye, suppose consider this is the shape of the eye, this much. But once the mutation happened, 4 base pair deletion, the globe size is very low. You can compare the globes, because he is working in the previous speaker, he is from Korea. So you like that, you have to connect it. The ஒரு ஜோக் கூட இருக்குல்ல, ஒரு ஸ்டாம்பை கொடுத்தா அவன் வந்து ஏதோ ஒரே ஒரு ரூபாய்க்கு தான் ஸ்டாம்பு கொடுத்தான், அவன் இங்கிலாந்து ராணி பற்றி பேசுகிறான். இது ஒரு ஜோக் இருக்குல்ல, சரத்குமார் அண்டு தட் வடிவேல், ரைட்? ஏன்னு ஒரு படம் இருக்குல்ல, அது பார்த்தீங்களா? போய் பாருங்க நீங்கள். Okay, so even a single thing, you have to connect it, build up many things. For that you need more information. For that you have to read in deep. Okay, so I am comparing the globe of the Asians, especially in Indians and Koreans, because the globe size is very small when compared to us. Okay, the same way here, if there is any 4 base pair deletion, the zebrafish, see here. So this is the wild, I think we have to turn off the light. See here, the globe, okay, so this is the normal fish, the globe is big and the globe is reduced here. This is the mutant, 4 base deletion. You compare this globe and this globe. Okay, so like that, if you go inside, if you go to the inside of the retina, and what are the structure is reduced. Okay, I don't want to go in detail what are the layers and everything, the flexiform layers and everything. So that is completely reduced. It means that transcription factor SIX playing a major role for glaucoma. That is the conclusion. All right, next slide. So next, the aniridia, again that is the name of the disease. Here, next slide. So here the gene is PAX6. So I told you 3 genes. One is FOXL2, another one is the SIX6, the third one is the PAX6. All the 3 are transcription factors. Okay, so that is relevant to the conference. So again here, this is the gene, and then what Nidhirajan did. So those, please, go back to the future. Okay, 2000, in the year 2000, just completed the human genome. There is no sequencer in India. No sequencer. So what we did, we all 3, what we'll do, either we'll do sequencing ourselves, manual sequencing, or SSCP analysis, or we'll send the sample to Korea, Switzerland, and then we'll do the sequencing. Okay, no sequencer in India at all. Even those days, we did the human genome sequencing in our lab. Okay, then now we have high-end machines. We have 4 sequencer, we have MySeq, so many sequences we have in our lab. Those days, with a limited source, but you have to thinking small is crime. You have to think big. Those days, we thought big, and then that's what he published more than 4 or 5 papers all together. Okay, so again this is the PAX6. Next slide. Okay, so if you are really interested, again these are the publication of Nidhirajan. You can see here are the 3 papers. See here, Nidhirajan, Nalathambi, myself. I'm the guide, so obviously I'm the corresponding author. So all the 3 papers. So here, what emphasize, nobody reported those days what is the genetic variation for aniridia. Again, aniridia, you can ask a question. Somebody I told you, sir, you told about that narrow eyelid, and then you told about glaucoma. What is aniridia? So aniridia, in the sense, iris, iris is a part of the lens. If there is any mutation in the PAX6, this is a complete iris, it is like a mild moderate severe, sometimes full iris, sometimes partial, sometimes only the rudiment. But see, you are normal. That's what you don't know the suffering of the patients. In America, they have an association. All the 3 diseases, aniridia, they have an association. No, we interacted and got some, tried to get some fund. Okay, Anaridia Association. Because the parents, you know, they have to give some good, like a right, positive words to their children. Because they have a Anaridia. Okay, they formed a group and then they discuss, is there any improvement? So where is the treatment? Okay, we are getting lot of emails, you know, once you get published this paper, they will write, you know, I think my son is having problem, what to do, how to avoid this, I have to understand my sequence. Because right now, people have money, but they are not, I mean, taking care of their health. Again, I am telling you, I am giving a message, if you want to live healthily, happily, please, I mean, understand your own genome, change your lifestyle, live longer. That is the message. Next slide. Next slide. Okay, I think maybe you guys are, I mean, mentally tired because I'm giving more information. Sometimes, you know, for some people, whatever I'm telling, it will go into your ear, it will stick on your brain. For some many people, you know, it will flying overheads. Okay, it is flying overheads. Okay, let him talk, you know. Let him talk. It is flying overheads. Okay, I will conclude like this. See, Nidhirajan, you know, and Nalathambi, long back in the year 2000, when before Dr. Abdul Kalam became a president, he was the scientific advisor to the Prime Minister, Vajpayee. Okay, that time he visited our lab. You know, we established our lab. So I think, see here, Dr. Abdul Kalam, the Nidhirajan, head of the department, Nidhirajan. See the young, handsome guy here. Who is this? Me. Hey, you can't able to believe me. I have to dye my hair, you know. This is me. Next slide. And every Thursday we have a lab meeting. You know, please, at least, you know, there are a lot of trees in our garden, in our campus. Form a group, eat lunch together, discuss something, you know, related to subject. Don't gossip, don't, you know, rubbish, don't be rubbish. Okay, discuss something, you know, please. Okay, every Thursday we have a meeting. See, this is again Nidhirajan and this is me, and this is a very well-known immunologist in India, Dr. Muthukarpan. See, again, you know, there is a lady section and a gent section, you know, we put it that way. Next slide. And Nalathambi, yeah, Nalathambi is, Nalathambi is here. This is Nalathambi. And this is Nidhirajan. Even, you know, our lab, see, I am collaborating with many countries. Okay, except Africa, you know, I visited all the countries, all the labs I know. I given many talks in more than 20 or 30 universities in US. So I think this is he is the director of SERI, Singapore Eye Research Institute. We went to many conferences to Singapore. So he is working on myopia. You know what is myopia? Why people are wearing the specs? This is a progressive, because aging. I'm reading glass. Okay, it's a myopia. So he is working on myopia. If there is any near work, you know, obviously problem in the eye, they have to put them in thick lens. Okay, so the Singapore, you know, they are pouring lot of money for myopia research. Okay, so he visited our lab those days. You know, I think this is the kind of, I mean, scientific interaction we had. You know, that's, I am selecting. I have millions of pictures in my laptop. I think I picked up very few. Okay, next slide. Okay, I think as I told you, I have more than 25 PhD students. So I think these are my PhD students already graduated. Most of them are settled in the US. I think Nidhirajan and Nalathambi is here because to teach you well. That's what they are here. They have sacrificed their life for you. Yes or no? So read please follow his instructions, read well. Okay? These are my PhD students. Next slide. Next slide. Thambi, adutha. Okay, I think I got many awards. Even I got the 2 awards from, this is the award from President of India. I went to the Rashtrapati Bhavan, I got the huge award. Okay, and this is Tamil Nadu Scientist Award, and another 2 awards from the Health Minister. That time the Congress government, Ghulam Nabi Azad was the Health Minister. I got those awards, and Dr. Abdul Kalam, when he was the President, a farmer, and after that, he inaugurated our institution, AMRF. That time, he came and interacted with us. This kind of interaction, what he asked me, sir, right now the human genome project is completed. People are asking my genome. The cloning strategy. The cloning is, is another, I think that is another strategy. The cloning is not, ethical. Here, I think, suppose story that, you have seen the Jurassic Park, the movie, Jurassic Park. They clone the, yes or no, Jurassic Park. So Jurassic Park, those days, the mosquito got the blood from the dinosaur, and then it has become fossil. Okay, they have taken the blood, isolated the DNA, and then they have taken the DNA and then they put it in the frog egg. Okay, like that, they developed the dinosaur. I'm talking about that book and then story. Okay, they can able to produce only the female dinosaur, not the male dinosaur. English padam paarenga thambi, Tamil padam paakadinga, English padam paarenga. Okay. Okay, so like that, he asked what kind of can I able to give my DNA to them. That kind of discussion is going on with the president, former president. Next slide. I think this is my current students. Like they left long back, still I have students, they are working for us. Okay, I think I can stop here. If there is any question, I will ready to answer. Next slide. Thank you. இது என்னது இத தான் சொல்லிட்டு நான் இல்லையா yes so what is this yes it means you can because you think you can என்னால முடியும் அப்படினா முடியும் today is not the end of the world இன்னைக்கே வந்து உலகம் அழிஞ்சிர போதா அழியாது it will take long time to ஒரு காலத்துல இன்னொரு கோடி கணக்கான ஆண்டுகளுக்கு பின்னாடி சூரியனே இருக்காது அது வரைக்கும் நாம இருக்க மாட்டோம் that is different story okay so today is not the end of the world please you can because you think can okay I will stop here if you have any question I already told you write in the question in the notebook if you have any question I am ready to answer thank you
hello sir so FOXLA gene you said about FOXLA gene So there is a probability in the first trimester the mother will be tested for
okay super
the mother will be tested for syndromes especially chromosome number 21 Down syndrome and all can be checked. Is there a possibility that we can check FOXLA gene also?
Okay sorry okay talking about the prenatal diagnosis
yes correct
அப்படினா என்ன தெரியுமா ஒரு அப்பா நார்மலா இருக்காங்க அம்மா நார்மலா இருக்காங்க ஆனா பிறந்த குழந்தை வந்து டிஃபெக்டிவா இருக்கு இப்போ என்ன பண்ணுவாங்க ரெண்டாவது குழந்தைங்க நார்மலா பிறக்கணும் அப்படினு ஒரு கியூரியாசிட்டி இருக்கும் எஸ்பெஷலி மாமியாருக்கு இருக்கும் இல்ல yes or no so in that case what we supposed to do we have to sequence the father's DNA mother's DNA and then see the exact where the mutation is and the mutation வந்து the affected child வந்திருக்கா அப்படினு பார்க்கணும் okay so now 3 pictures father mother and then affected child இப்போ இவங்க என்ன கேக்குறாங்கன்னா this is sperm this is egg fused together form a zygote புரியுதா once zygote ஆயிடுச்சுனா அவ்வளவுதான் ஓகே, ஒன்றும் பண்ண முடியாது. ஓகே, அதை எப்படி டெஸ்ட் பண்ணலாம்னா, ஒன்ஸ் தே கன்சீவ், ஓகே, கன்சீவ்ட் ஆர் 4 weeks, ஓகே, ஆம்னியாட்டிக் ஃப்ளூயிட், ஓகே, ஆம்னியாட்டிக் ஃப்ளூயிட். இப்போ நம்ம வந்து எங்கள் ஹாஸ்பிட்டலில் எடுக்க மாட்டாங்க, சென்னையில் போய் யூ சப்போஸ் டு டேக் த ஆம்னியாட்டிக் ஃப்ளூயிட் இன் a ஸ்மால் வால்யூம், தட் இஸ் கால்டு, நிறைய படிங்க தம்பி, நான் ஒரு வார்த்தை சொன்னால் நீங்கள் ஒரு புக்கில் இன்ஃபர்மேஷன் இருக்குது, தட் இஸ் கால்டு லிக்விட் பயாப்சி. Liquid biopsy. That the liquid biopsy, no, from that you can take the DNA and then அவங்களுக்கு ஆன்சருக்கு சொல்லிடலாம். ஓகே, இந்த குழந்தை, இந்த கன்சீவ் ஆன குழந்தை மறுபடியும் வந்து டிஃபெக்டிவாக பிறக்க போதா நார்மலாக பிறக்க போதானுங்கிறத நம்ம சொல்லிடலாம். ஆனால் அந்த ஃபாதர் அண்ட் மதர் தே ஹாவ் டு டிசைட் தெம் செல்ஃப் அபார்ட் பண்ணலாமா. சப்போஸ் அது மறுபடியும் அதே டிஃபெக்டாக இருந்துன்னு வச்சுங்க, அந்த அப்பா அம்மா தான் டிசைட் பண்ணோம் அபார்ட் பண்ணலாமா வேணாமான்னு. But if the scientist, what they will do, this is the scenario for the baby அப்படின்னு சொல்லலாம். ட்ரைசோமின்னு ஒன்று இருக்குது மேடம். she is talking about the chromosomal aberrations. நம்ம ஹியூமன் குரோமோசோம் எத்தனை தெரிஞ்சவங்க மட்டும் சத்தம் போடாதீங்க, கை தூக்குங்க பார்க்கலாம். ஹியூமன் குரோமோசோம்ஸ், சூப்பர், எக்ஸலன்ட். உங்கள் பேர் என்னம்மா? திவ்யா. she is the right answer. 23 pairs, 23 pairs, உட்காருமா, 23 pairs, 22 autosomes and then XY chromosome. 22 autosomes, you XX, I am XY. That's what I am male, you are female. Okay, so totally 46 chromosomes there. சரியா? அப்புறம் உடனே மைட்டாசிஸ்க்கு வாங்க. மைட்டாசிஸ், when the cell divide, equal number 46. if it is a மைட்டாசிஸ் தான் சொன்னேன். so மியாசிஸ்ல எப்படி இருக்கும்? the n number is 1n, 2n for mitosis, n for meiosis. அப்பனா 23 தான் இருக்கும். எக்ல 23, ஸ்பெர்ம்ல 23, ரெண்டும் ஃபியூஸ் பண்ணா நார்மல் சைகோட். sometimes that is called chromosomal aberrations, chromosomal autogamy அது சொல்றாங்க இல்லையா. so சில சமயத்துல chromosome நம்பர் 1 வந்து 2 இருக்கும், diploid இருக்கும், அது like that. if it's so the trisomy madam, trisomy. so if it is a trisomy, obviously the baby having some problem. Okay, the baby born with some defect or syndrome, not only problem in the eye. இப்போ இவர் நீதிராஜன் ஒர்க் பண்ணார் இல்லையா, அந்த அனரீடியாவிலே 3 டைப் இருக்குது. ஒன்று கண்ணில் அஃபெக்ட் பண்ணும், ஒன்று கிட்னியில் டியூமர் வரும். சில குழந்தைங்களுக்கு ஐலையும் ப்ராப்ளம் இருக்கும், கிட்னிலையும் டியூமர் இருக்கும். சில குழந்தைங்க ஐயில் ப்ராப்ளம் இருக்கும், மென்டல் ரிட்டார்டேஷன். everything I think is a problem in the gene or the chromosomal aberrations. okay, thank you Divya, at least you asked the right answer, a right question. யாருங்க உங்களுக்கு வாத்தியாரு? ஒரு 5 மார்க் அதிகமாக போடுங்க இந்த செமஸ்டர்ல. oh I see, you are Ferrier. I think I know the zoology department. what is her name? okay, I think she did PhD in with Gunasekharan. Okay, zoology நீங்க? சரி. Yeah.
So what kind of method you used to inject the GPROFISH model gene injection?
Okay, okay. It's called micro injections. இப்போ நீங்க நமக்கு ஊசி போடுறீங்க இல்லையா, அது எத்தனை காஜ் நீடில் தெரியுமா? அதெல்லாம் தயவு செஞ்சு நீங்கள் மிக்சர் சாப்பிட்றீங்களா, மிக்சர் அது பேப்பரை தானே கட்டித்தரான், சாப்பிடுங்க. அந்த தூக்கி போடுறதுக்கு முன்னாடி அந்த பேப்பரில் என்ன எழுதிருக்கான்னு சொல்லிட்டு தூக்கி போடுங்க. த சேம் வே தட் காஜ் நீடில் 23 காஜ் நீடில் ஆர் 25 காஜ் நீடில், தட் இஸ் நத்திங் பட் த டக் இருக்கலாமா, அந்த டயாமீட்டர் உங்களுக்கு தெரியணும். ஓகே, வித்தவுட் அஃபெக்டிங் தட் பர்டிகுலர். அதுக்கு வந்து அமெரிக்காவில் இருக்கும்போது ஐ ஒர்க் வித் பிரெயின், அதெல்லாம் பெரிய பெரிய ஸ்டோரி. நீங்கள் என் பேரை மட்டும் அடிங்க, பப்மெண்ட் தெரியுங்களா, பப்மெண்ட்டில் என் பேரை அடிங்க. ஸோ லைக் வி ஒர்க் வித் பிரெயின் டியூமர். அந்த பிரெயின் டியூமரை க்யூர் பண்ணுறதுக்கு ஒரு கேன்சர், அதை வைரஸ் வந்து உள்ள தள்ளணும், அந்த பிரெயின் டியூமரில் உள்ள தள்ளணும். அப்படிங்கும் போது இந்த மாதிரி டீட்டெயில் தான் வி யூஸ்ட். ஸோ தட் இஸ் கால்ட் மைக்ரோ இன்ஜெக்ஷன். கன்சிடர் திஸ் இஸ் அ ஃபிஷ் எக் and then you supposed to push that the plasmid into the egg. இல்லையா? அந்த பிளாஸ்மிட் வந்து அஃபெக்ட் ஆகக்கூடாது. அதுக்கு தகுந்த மாதிரி அந்த டயமீட்டரை வச்சு நீங்கள் வித்தவுட் டேமேஜிங் த எக் புஷ் இட் இன்சைட். யூடியூப் பாருங்களாங்க, கண்ட கருமமும் அந்த இதில் யூடியூப்பில் பார்க்குறதுக்கு சயின்டிஃபிக்காக ஏதாவது பாருங்கள். ஆல் இஸ் தேர். ஓகே, இல்லை she is asking, see, there is a candidate gene. One, if it is a candidate gene in the sense, this particular gene, if it is mutated, it will form the small eye. That is called candidate gene. That is some gene for breast cancer, someone for this one eye disease, like that. One particular gene that is called CCT, that is name of the gene, very recently identified. You take that gene, you make a mutation, and then you take the mutant, push it into the egg, and you grow the egg. Then that the fish, it will defect in the eye, globe size. Yeah. Very good. இல்லை, இதெல்லாம் வந்து இவங்க கேட்குற கேள்விலாம் இட்ஸ் அ வெரி வேலிட் கொஸ்டின். இப்போ ஹலோ தம்பி, கொஞ்சம் பேசாமல் தம்பி ப்ளீஸ், நான் கொஞ்சம் பேசுகிறேன், அப்புறம் நீங்கள் பேசுங்க ப்ளீஸ் தம்பி, கொஞ்சம் அப்புறம் பேசுங்க தம்பி ப்ளீஸ். அதாவது ஐ திங்க் சி இஸ் ஆஸ்கிங் an excellent question. ப்ளீஸ் உட்காருங்க. வாட் இஸ் எத்திக்ஸ்? எத்திக்ஸ்னா என்னன்னு தெரியுமா? நான் பாட்டுக்கு சும்மா இப்போ மவுஸெல்லாம் கொள்றோம்ல, இப்போ இவங்க கேட்ட கேள்வி அருமையான கேள்வி. இப்போ நான் அதில் நிறைய வேலை பார்த்துருக்கேன். கன்சிடர் த மவுஸ். ஒரு டியூமர் வருதா வழியானுக்கு இப்போ பண்ணுறதுக்காக யூ டேக் த டியூமர், பிரஸ்ட் கேன்சர் செல்ஸ் ஆர் யூட்டிரஸில் நிறைய கேன்சர் வரும், யூரினரி பிளாடர், ப்ராஸ்டேட் கேன்சர். அந்த செல்லை வந்து இன்விட்ரோவில் அந்த செல்ஸை எடுத்து நியூட் மவுஸ், இம்யூனாலஜி நியூட் மவுஸ்னா அந்த அந்த டியூமர் செல்ஸை வந்து இங்கே இன்ஜெக்ட் பண்ணுவாங்க. வித்தின் 15 days it will form the tumor. அந்த டியூமர் வந்து ஒரு பர்டிகுலர் சைஸ்க்கு தான் இருக்கணும், அதுக்குள்ளே நீங்கள் அந்த அனிமலை கொன்னுடணும். ஓகே, நான் எதுக்கு சொல்கிறேன்னா எத்திக்ஸ். அந்த 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 ஏற்கனவே அந்த அனிமலை நீங்கள் வந்து இது பண்ணுறீங்க, தொந்தரவு பண்ணுறீங்க. அந்த டியூமர் பேர்டன் இருக்குல்ல, சில சமயத்தில் அப்படியே அதை தூக்கிட்டு நடக்க முடியாது, அதனால் சாக்ரிஃபைஸ் பண்ணிடுன்னு சொல்லுவாங்க. அதனால் மவுஸுக்கே இப்படி பண்ணும்போது ஹியூமனுக்கு எத்திக்கல் கமிட்டி வாங்கணும். நீங்கள் ஒரு கிராண்ட் எழுதுறீங்க, ரிசர்ச் கிராண்ட், ஐசிஎம்ஆர் ஆஸ்கணும் இந்த எத்திக்கல் கிளியரன்ஸ். இது எது? நீங்கள் செல் கல்ச்சர் பண்ணுறீங்களா, ஹியூமனை டச் பண்ணுறீங்களா, டிஎன்ஏ எடுக்கிறீங்களா, வெதர் இட் இஸ் இன்வேசிவ், அங்கே இன்ஜெக்ட் பண்ணுறீங்களா, யூ ஹேவ் டு ஆர் யூ கேன் கெட் த கன்சர்ன் ஃப்ரம் த பேஷண்ட். நான் ஒத்துக்கிறேங்க, எல்லாம் நீங்கள் என்ன பண்ணுறீங்களோ ஒத்துக்கிறேன்னு சொல்லி கையெழுத்து வாங்கிட்டு தான் பண்ணணும். We have an ethical committee. We have proper channel to do the research. Very good question. Yeah, I think very recently they modified. See, I am in many committees in government. Okay, so she is asking. Previously, again we can classify the animals: small animals, big animals. In America, they are using monkey, pig, dog, goat. Okay. So here people are using only mouse or zebrafish. Previously there is no clearance for zebrafish. Now the DST or the DBT, they told definitely, even at least you had to get the consent from the committee. Yeah.
The story starts back in the mists of time, the early 1990s, when the gene for an eye condition called aniridia was identified. Here's that. I don't know if you can see at the bottom of my screen here. There is a little baby that's been born with aniridia, which means absence of the iris. So when the baby is born, the doctors notice that the eye doesn't have an iris, they've just got big black pupils. And it's transmitted in an autosomal dominant manner. And as you can see by these papers that are up on the screen here, they were identified, the PAX6 gene was identified as the candidate gene for this condition. As I'll go on to describe, PAC6 is encodes a transcription factor, which you can see in the top right of the screen here is expressed in brown throughout the developing eye, in the lens epithelium, the developing retina, and the epithelium over the top of the eye, the corneal epithelium. In the lab that found the human gene, this was in Edinburgh in the early 1990s, the lab next door identified that a mouse mutation called small I was also caused by heterozygous mutation in the mouse's home. Down at the bottom here are some embryonic mouse embryos, embryo 14, and is a PAC6 homozygous mutant, and you can see it doesn't have any eyes. And the heterozygous in the middle, it does have eye, but the eye is small, and hence it got called the small eye phenotype. I mention this because people my age and younger and older, sorry, it wasn't obvious at the time that human developmental transcription factors could cause to all the young people out there, this is now kind of that PAC6 was probably the first developmental transcription factor that was linked to a human genetic disorder. And that's why these papers are in Cell and Nature, because at the time it was really new, it was a really big deal. And then the intense research into PAC6 and related transcription factors. And one of the key results was that PAC6, this transcription factor, has an evolutionarily conserved role in eye development in pretty much every animal that has eyes. So, for example, on the left of this screen, you see a picture I've already showed you of the PAC6 mutant mice. And on the right, in the mid-1990s, the gene was identified that causes the Drosophila ileus mutation. Yes? Oh, okay. Sorry about that. I thought I was sharing. Okay. So okay. A cup-like item. Hold on. This is a shame. Hold on. So you can't see. Okay, but it's not in presentation mode. Okay. Can you see now? I don't. Yeah, okay. Is that better? I don't have that right now. What can you see right now? Yeah. Okay, good, good. Yep. I don't see an ad. Yeah. I don't see that, no. I don't have that. Okay. I mean, I can present like, if you can see my slides like this, I can present like this. Mm-hmm. I don't have anything that says add notes. That's not what I'm looking at the moment. Hold on. Okay, so yeah, yeah, that's what I'm seeing. Yes, yeah, yeah. I don't have anything that says add notes. Uh, I don't have that. I don't know. Right. That's what I'm trying to do. So wait, well, when I do that, can't you see the presentation mode? Yeah. No, that's yes. Okay. This is very strange. I wonder what's happening. I'm doing that just now. Can you see now? Okay. Hey. Okay. Good. Yeah. Right. So sorry about that, everybody. I'm afraid it's an old people problem with computers. Right. Let's go again. So I'll briefly recap the stuff that you couldn't see, and then I'll keep going. So this slide you couldn't see. You missed the important bits. Here's a little baby born with aniridia. Very cute. Doesn't have an iris. This is expression of the PAX6 gene in the developing eye in brown. It's pretty much everywhere, retina, lens, and cornea. In 1991, a paper was published showing that the PAX6 gene was the cause of human aniridia. And the lab next door found that the PAX6 gene was the cause of a human mouse mutation called small I, where the mice also don't have an iris. The next slide that you couldn't see was that the evolutionarily conserved role of the transcription factor PAX6, and showing that here's some mouse embryos, embryo at day 14, a normal embryo, a PAX6 mutant embryo that doesn't have any eyes, and a heterozygote which does have an eye, but it's a small eye with aniridia, and the Drosophila iris gene as it was discovered in 1995, is also caused by mutation in the fly's homologue of the PAX6 gene. And pretty much what that means is that PAX6 has an evolutionarily conserved role in eye development, such that if you're an animal with eyes, then you are using your homologue, your orthologue of the PAX6 gene to build your eyes. And that's a whole new different story about that, how that happens, which I won't go further into today. But in terms of mammalian development, you can see if you don't have any PAX6, you don't have any eyes. And unfortunately, you're also dead. It's an important brain gene. Mice and people born with homozygous mutations in the PAX6 gene don't have quite severe brain abnormalities and a lack of nasal epithelia, which is incompatible with life. But heterozygotes do survive, but they do have eye problems. So that's what this slide. As I've already said, PAX6 people don't have any eyes, zygous mutant, and that's because the genes required for some very early stages of eye development, which include lens induction, the development of the retina, and the development of the cornea. Heterozygotes for PAX6 are perfectly normal people, but they do have quite severe eye problems, which include, as I've said, aniridia, the absence of the iris, as you can see in the bottom right here. They are predisposed to cataracts, glaucoma, and foveal hypoplasia, spelling mistake, foveal hypoplasia, so that the retinal visual acuity is not great. And they also have corneal opacity, so-called aniridia-associated keratitis. And in terms of what it means, the eye is exquisitely sensitive to PAX6 dosage. Essentially, any less than about 70% PAX6 activity, or any more than about 130% of normal PAX6 activity, and you start to get eye defects at birth, which is, again, it's a really interesting phenomenon. And it's a really good example of a tissue, the eye, in particular the lens, being very, very sensitive to dosage of a transcription factor. Most of what I'm talking about is this aniridia-associated keratitis, which you can see on the right-hand side of the screen here, where the cornea, the front surface of the eye, becomes inflamed, vascularized and opaque, and it occurs as the kids with aniridia are getting older. So when the babies are born, they don't have an iris, obviously, and they are predisposed to cataracts as they get older. And the visual acuity is never that good because of retinal problems. But all these things don't really affect their lives. You can have cataract operations, you can wear sunglasses for the aniridia. The problem is, as these kids get older, this corneal opacity starts to take hold, such that by early adulthood, many of them are registered blind. They can see light and dark, and they may have some visual acuity, but most of them are effectively registered blind. And this is the unmet clinical need, because there really is no effective treatment for this. Corneal transplants don't work in aniridia patients because they appear to have very poor wound healing capacity. And it's very risky to operate on an aniridic eye anyway because they don't heal well. And this is the problem that we've spent most of the last 20 years trying to rectify, or trying to understand what's happening so that we can rectify it. And the fact that it's taken over 20 years, I think will show you how difficult it is to move from knowing what the gene disorder is to explaining what the clinical phenotype is, the so-called phenotype gap. It's very good to find the gene mutation that is causing a human genetic disorder, but it's a long way from being able to understand how that gene mutation causes the disorder, the mechanisms, and therefore what you can do about it pharmacologically. So stepping back a bit, I'm going to describe the cornea a little bit for those of you out there who aren't very familiar with it. Top right of the screen here is an eye. You're all familiar with that. We see them every day. And the cornea is the so-called window on the world, the transparent structure at the front of the eye with the iris lying behind it, and the whites of the eyes, the conjunctiva, and the sclera surrounding it. And if you take tissue sections through this, okay, this is a mouse eye, but it's got the same bits as a human eye. The cornea is a collagenous structure. Most of it, the stroma, is very hypocellular, not much in it, but there is an epithelium running over the top, a stratified epithelium that expresses PAX6. And the cornea is really only a specialized skin. It's a transparent skin, it's a simple skin, but it is a specialized skin. And you're aware that your skin contains stem cells that are essential for maintenance of the epidermis of the skin. And sure enough, the cornea contains stem cells that are essential for maintenance of the cornea. And these stem cells actually live around the edge of the cornea. At the boundary between the cornea and the conjunctiva, the so-called limbal ring, there's a population of stem cells there, limbal stem cells, that are responsible for maintaining the cornea. Obviously, it's difficult to do experiments in people and in human tissue, but luckily the PAX6 mutant mouse, the heterozygous mouse, is a really good model of human aniridia-associated keratopathy. On the screen here, on the left is another PAX6 heterozygote, the so-called small eye mouse. You measure them, they're not much smaller, they're about 5 to 10% smaller than the wild-type mouse. But the PAX6 mutant mice also have aniridia and cataracts and the keratopathy, which very faithfully recapitulate the human aniridia phenotype. Now the stem cells, the limbal stem cells, are detectable in mice as so-called label retaining cells, and I will explain briefly what that means. Being a specialized skin, cells in the corneal epithelium are dividing by mitosis all the time. As it turns out, that the stem cells divide much more slowly than the other proliferating cells in the cornea. And what that means is that you can label tissues in the mouse with thymidine analogues such as bromodeoxyuridine. BRDU is a chemical that you've probably heard of, that it acts as a thymidine analogue and will incorporate into replicating DNA. And what that means is that if you label a mouse with BRDU by putting it in drinking water or by injecting BRDU, all the cells that are dividing will incorporate BRDU into their DNA, and then you can detect that later by using antibodies in tissue sections or in whole mount tissue immunohistochemistry. And stem cells are label retaining because they are very slowly dividing, so that once they contain BRDU in their nuclei, they divide very slowly, and it dilutes out very slowly. Whereas a rapidly dividing cell, such as in the skin or or over much of the ocular surface will lose the BRDU really quickly because it's dividing all the time. It dilutes out the BRDU. So this slide just comes from an oldish paper that was labeling postnatal mice with BRDU, in this case for 4 days. And the BRDU labeling is shown in magenta. And you can see that after 4 days of BRDU labeling, nearly all of the cells in the ocular surface are labeled with BRDU. But then if you do a 4D BRDU label and then take away the BRDU, just give the mice normal drinking water for 9 weeks, most of the BRDU dilutes out, but you see a few label retaining cells identified by arrows here and there in the ocular surface. And what this paper showed, as I'll show on the next slide, if you map out where the label retaining cells are, this is a flat mount cornea, they're all around the edge of the cornea in the limbal ring. So this is how you identify that these stem cells are present in the limbus, because the only place where you find label retaining cells is the limbus. Okay, so that's old work, really, demonstrating how we identify the presence of stem cells in the limbus. And you may be asking, well, why am I telling you all this? What's the point? Okay, it's a good question. But the reason is it's relevant to aniridia-associated keratitis. What you're seeing on the screen here, this beautiful mouse eye that looks a bit like a sweet, is the result of an experiment that we did several years ago that was looking at patterns of cell maintenance in the mouse cornea. This is a female mouse who has an X-linked LAC-Z transgene. Now, LAC-Z encoding beta-lactosidase allows you to stain cells blue. And this female mouse has one X chromosome carrying the LAC-Z transgene. And because of random X inactivation during development, some of her cells are blue and some are white. And in most tissues, it's a horrible patchwork mess of blue and white cells. But in some tissues, it's not. And here you're seeing the corneal epithelium. And what you're seeing, these stripes of cells, is that in the limbus, around the edge of the cornea, there are patches of blue and white stem cells, and they are producing streams of blue and white corneal epithelial cells that are migrating into the cornea. Think about it. Every time you blink or get a bit of dust in your eye, cells fall off the corneal epithelium. Those cells have to be replaced, and substantially they're being replaced by stem cells around the edge that are producing streams of new cells that migrate into the cornea to replace those that have been lost. And this is actually essential for normal corneal maintenance. If the stem cells die, either by wounding or by, for example, alkali burns, industrial accidents, for example, then the cornea can no longer be maintained. You lose epithelial integrity, the cornea becomes opaque and effectively and gets scarred, and people can effectively go blind. This horrible looking eye that you see on the screen here, I think, is a result of an alkali burn, I think. And the point is that aniridia-associated keratitis looks an awful lot like a limbal stem cell deficiency. Again, here is this is a 12-year-old kid with aniridia-associated keratitis, and the vascularization, the inflammation, the opacity, the corneal scarring and pannus looks like a limbal stem cell deficiency. And historically, and even today, aniridia-associated keratitis is treated clinically as a limbal stem cell deficiency. And the question is, one of the questions is that really true? Is that what it is? So we started a series of experiments to try and look at corneal maintenance in the PAX6 mice. And we did that by breeding PAX6 heterozygous mice with the blue mice carrying the X-linked LAC-Z transgene, so that we would get from these litters blue and white mice that were either heterozygous for PAX6 or homozygous wild type for PAX6, and we could compare them and see what we got. And the kinds of results we got are on this slide here. Here is a wild type eye with the blue and white striping that you expect, which, as I've said, reflects the migration of cells from the limbus into the cornea. And here are a variety, these three panels are sort of representative of the sorts of patterns that you get when this LAC-Z transgene is on a PAX6 mutant background. And you can see it's really variable. You can get eyes on the right here that look more or less normal. You can get eyes on the left here that look very badly messed up. There is really no evidence of striping patterns. But most of them are somewhere in between, where you see patterns like this, where you can see evidence of a striping pattern, but it seems to have been badly messed up. So there is variability in corneal maintenance in the PAX6 mutant mice, which, as it happens, reflects the clinical variability in the disease. But clearly having PAX6 mutation doesn't preclude you from having reasonably normal levels of cell migration and presumably limbal stem cell activity in the cornea. Again, back in the old days, we made chimeric mice by aggregation of PAX6 mutant embryos and wild type embryos in vitro, and then reimplanting into pseudo-pregnant female mice in order to get chimeric mice, which are the things that you see here, with variable mixtures of wild type and PAX6 mutant cells, all very little nice cute mice. But we looked at their eyes, they were carrying LAC-Z transgenes as well, and we get this sort of pattern. Now, the difference between these eyes that you're seeing here and the eyes that were on the previous slide was that in this slide, the white and the blue cells can be different genotypes. So here's a control eye with the white and the blue cells are both wild type, and you see normal patterns of corneal striping. The other three are eyes where the blue cells are PAX6 heterozygotes, but the white cells are wild type. And you're seeing relatively normal patterns of striping. So, for example, top right here, all these blue cells here are PAX6 mutant cells, and clearly the PAX6 mutants can form patches of mutant stem cells which migrate normally to the center of the cornea. So, given some wild type support, PAX6 mutant cells can behave perfectly normally in the cornea. And it's wrong to suggest that PAX6 mutant cells can't form limbal stem cells and can't migrate properly, because clearly they can. Some of the quantitative stuff we've done suggests that it may be that fewer clones of limbal stem cells are specified in PAX6 mutant mice, and that mutant stem cells are not as competitive as wild type in forming limbal stem cells. But PAX6 mutants do have stem cells, and these stem cells are capable of maintaining the cornea during homeostasis. So if there is a problem with these stem cells, what is it? And the other question that we addressed therefore was wound healing, because the dogma about these stem cells in the cornea, as in many other tissues, is that during normal tissue homeostasis, they aren't doing very much, but they divide relatively slowly, maybe only every 10 to 14 days, just enough to maintain those striping patterns into the cornea. However, when the cornea is wounded, so a scratch wound or any other sort of injury, it has been assumed that the limbal stem cells start to divide more quickly. So that if there is an acute injury to the cornea, the stem cells will divide much more quickly and produce more cells for maintaining the cornea. And although that had been the accepted model for since the stem cells were identified 30 years ago, nobody had ever really proved that that was the case quantitatively. So a few years ago, I sent one of my Nada Saga onto the question of how do these stem cells work, and she did some work in wild type mice, saying, do limbal stem cells respond to corneal injury? And she did an experiment where she gave normal wild type mice a thymidine analog. She didn't use BRDU, she used iododeoxyuridine, IDU, because it's cheaper actually. She gave mice IDU in their drinking water, and then she stopped it and gave them 11 weeks of normal drinking water, such that all rapidly dividing cells would dilute out their IDU, but slowly dividing cells, such as the limbal stem cells, would retain some of their IDU. After that period of time, for each mouse, she injured one eye. So she did an epithelial scrape wound. Under anaesthetic, she scraped off about a millimetre circle of epithelium from the cornea, one of the corneas of the mice. And at the same time, she gave them a dose of another thymidine analog, chlorodeoxyuridine, CLDU, and then waited 24 hours, killed the mice, and then took their eyes for immunohistochemistry. And what she was looking for was double-labeled cells. Any label-retaining stem cells that had IDU, how many of them divided in injured and uninjured eyes in the 24 hours following a wound? And if the dogma was right, then what she would see is many more double-labeled cells in wounded eyes compared to unwounded eyes. This is just a representative picture of the sorts of things that she saw in the cornea. So here would be after injury, or with or without injury. So the red cell retained IDU, the green cells are CLDU, the cells that divided again in the last 24 hours of the mouse's life, and the blue is nuclear label. And she could visualize and count the proportion of cells that were IDU positive, that were CLDU positive, and were double labeled in injured and uninjured eyes. And to cut a long story short, this is the kind of stuff that she got. I'll really just focus on the right-hand side of this image, showing how many double-labeled cells were present in the limbus of uninjured and injured corneas 24 hours after the injury. And what you can see is that as we expected, there really was a very significant increase in the proliferation rate of limbal stem cells in injured eyes 24 hours after the injury. In uninjured eyes, only about 4% of limbal stem cells were dividing in 24 hours. In injured corneas, it was up to over 30%. Okay, so that's good. That's what we expected. One thing that we didn't expect, that we kind of found by the by, is that if she repeated the experiment giving the eyes, the mice IDU treatment, a washout, a corneal injury, but then left the mice for 48 hours and gave them another thymidine analog pulse in the last 24 hours. Then after 48 hours of wound healing, nearly all the limbal stem cells in injured eyes are dividing. But also, if you looked at the contralateral eye of every mouse, nearly half of them were dividing too. So after 48 hours, activation of limbal stem cells in one eye appears to activate the stem cells in the other uninjured eye as well, which we didn't expect. And I don't know why that is. Presumably, there's release of cytokines from the injured eye, which travel through systemically and may activate the stem cells in uninjured eyes. So we don't know why it happens, but it does happen. And here's a little cartoon that if you injure one eye of the mouse, then after 24 hours, the limbal stem cells have activated in that eye and are dividing to replace the cells that have been lost. But after 48 hours, both eyes are dividing. Nearly all the limbal stem cells in the injured eye and about half of them in the contralateral uninjured eye are activating in response to injury. And this is in wild type normal mice. So the obvious question is do we see the same picture in PAX6 mutants? And again, to cut a long story short, the answer is no. My subsequent student, Aklam Alamri, has been working on this problem in PAX6 mutant mouse. So here's her little picture. Here's a normal wild type mouse. Here's a PAX6 mutant small eye mouse. They both get the IDU labeling. They get the washout period. And then Aklam has been looking for double labeled cells in comparing wild type and PAX6 mutant littermates. And what she found, these are the sorts of images that she saw, but when she counted proportion of IDU positive cells, proportion of double labeled cells in PAX6 mutant mice before and after injury, she started to find a few interesting things. One thing, in normal, uninjured PAX6 mutant mice, the limbal stem cells are dividing faster than normal mice. So as before, in a 24-hour period, about 4% of limbal stem cells divide in wild-type mice, uninjured eyes, but in uninjured PAX6 mutants, it's higher. It's not a lot higher, but it is significantly higher. And how we interpret that is the PAX6 stem cells are having to work harder. What we do know is, as I'll come to, is that the PAX6 mutant corneal epithelium is fragile and cells fall off more quickly. So cells have to be replaced more often. And we think that the limbal stem cells are normally working that little bit harder than wild type in order to maintain the uninjured PAX6 mutant cornea. When Aklam did corneal scrape injuries of wild type and PAX6 mutant eyes, again she saw a difference. As expected, if you injure the wild type eye, limbal stem cells wake up and start dividing more quickly. Not the case in the PAX6 mutant eye. You injure the PAX6 mutant eye and look at stem cell activity 24 hours later, and it's the same. It hasn't improved. So the PAX6 mutant eyes, the stem cells either don't see the wound or they're incapable of responding to the wound. And to partially address which of those two options might be true, Aklam did the same experiment where she looks at cell proliferation 48 hours after 4 hours to improve. And this time she saw some really interesting stuff. That in wounded eyes, 48 hours after injury, suddenly the PAX6 mutant stem cells were awake, wild type. And as I've described, if you injure one, after 48 hours, the stem cells wake up in the other eye and start proliferating. And it was the same for PAX6 mutants. So in the contralateral unwounded eyes, after 48 hours, again, nearly all the PAX6 mutant limbal stem cells were dividing. So what we're seeing here is that there is a problem with PAX6 mutant limbal stem cells, but it's a delay in the response to wounding. It's not a failure of response to wounding. That PAX6 mutants do have stem cells, these stem cells can work, but they take longer to respond to wounding. And Aklam's little cartoon is now this, that here's the wild type situation. This is what I've already showed you. The PAX6 mutant mice with the smaller eyes, the only difference is at 24 hours. That in the wild types, the injured eye stem cells have activated, and in the PAX6 mutant they haven't, but after 48 hours there's no difference. The PAX6 mutant limbal stem cells can activate pretty much as well as wild type. That's a very subtle defect in the stem cells, and the question is, can that explain the clinical phenotype? That this very mild deficiency of limbal stem cell, is that enough to explain the catastrophic failure of corneas that we see in aniridia patients? In order to answer that question, we have to look at anything else that might be happening in the aniridic cornea. And another PhD student of mine, Jingxing Oh, really about 10 years ago now, was able to investigate that question. What goes wrong with, not with the stem cells, but with the actual corneal epithelium cells themselves, the central corneal cells. And one of his papers is up here. I mean, what he was able to show is that the PAX6 mutant corneal epithelium is a bit of a mess, and it's fragile, and it's lost its barrier function. So well, let's start with the nice pictures on the right here. This is a wild type mouse eye. This is a PAX6 mutant wild type PAX6 mutant eye, and you can see the opacity forming in the cornea. This opacity affects both the epithelium and the underlying stroma, and it's associated with a function of the mutant cornea. This is some scanning electron microscopy of mouse corneas, and the wild type corneas have a nice smooth barrier surface. The mutants, unfortunately, are not like that. Their epithelium is full of holes, and there are cells escaping from and flaking off from the surface of the mutant cornea. Because the cornea is full of holes, it loses barrier function. If you drop fluorescein on a mouse eye, normally it should stain only the very upper surface of the eye. If you drop fluorescein on a PAX6 mutant eye, you can see the fluorescein penetrates the whole cornea. So these corneas have lost their barrier function. Ooh, I'm getting some feedback now. What does that mean? Oxygen is getting into the PAX6 mutant epithelium. That predisposes them to higher levels of oxidative stress, and we've measured that directly and shown that the PAX6 mutant corneas are under oxidative stress. What the stress does is it causes the actual red color, the green color here is oxidative stress, and the red color is marks transdifferentiation of corneal epithelial cells into a kind of cloudy myofibroblast type phenotype. So that the epithelia is under stress, and that is causing dedifferentiation of the corneal epithelial cells, and that could explain some of the opacity. When we did proteomic studies of PAX6 mutant corneas, it appeared that they also had less of the protective heat shock chaperoning type proteins that wild type corneas normally have. Things like aldehyde dehydrogenase, transketolase, glutathione transferase. These are all protective enzymes which the PAX6 cornea is not producing as much of. Think of the problem. The cornea is under attack all the time from air, oxygen, pathogens, injury, blinking, eyelashes, and with the stem cell activity and protective biochemical pathways, normally it manages to survive. But it is on a knife edge, and any small thing going wrong, such as holes in the epithelium, loss of protective enzymes, appears to be able to tip the cornea over into a pathology. Why this affects PAX6 particularly wasn't immediately obvious, but Jingxing showed something else, which is that PAX6 itself is susceptible to oxidative stress. And what it's susceptible to is that oxidative stress drives it from the nucleus. So in a this here is a normal wild type cornea, and PAX6 in green should be expressed in the nucleus. The protein should be localized to the nuclei of the epithelial cells. In a PAX6 mutant, it's patchwork. Sometimes it's nuclear, but in other cells you find it's gone cytoplasmic. And Jingxing was able to show that is a response to oxidative stress. Because if you take normal wild type cells and put a low concentration of hydrogen peroxide on them, the PAX6 leaves the nucleus. You can see how that can exacerbate the PAX6 phenotype. Because if cells have a little bit less PAX6 than normal, that leads to oxidative stress, which drives the PAX6 that there is out of the nucleus. And remember, PAX6 is a transcription factor. It has to be in the nucleus to work. So what we have labeled this, because we're not serious, is the triple whammy model of aniridia-associated keratopathy pathology. That the aniridic corneal epithelium is fragile and it's got holes in it and it loses barrier function. In combination, it expresses lower levels of stress-responsive enzymes than it should do. And together what that does is that it leads to increased oxidative stress, which in turn expels PAX6 from the nucleus. And this leads in the PAX6 mutant to a chronic wound healing state and dedifferentiation. And the limbal stem cells respond to that by slightly increasing their proliferation, even in the apparently uninjured aniridic eye. And then this chronic wound healing state appears to delay the ability of the stem cells to respond to any subsequent acute wound. Hope for the future is that if we add reducing agents to PAX6 mutant cells, things like, beta-mercaptethanol, or a much more friendly reducing agent, glutathione, then we can restore some of these properties. But if we add reducing agent eye drops to PAX6 mutant mice, we can start to restore, we can push PAX6 back into the nucleus, and we can start to restore some of the wound healing. So this figure here is showing that adding reducing agents to PAX6 mutant cells restores their wound healing ability pretty much to wild type levels. These are control cells, cells with hydrogen peroxide where everything gets worse, cells with beta-mercaptethanol and glutathione where everything gets better, and this is a control dimer of glutathione that shouldn't have any effect, and thankfully it doesn't. Now, okay, Jing Xing produced this summary diagram of how PAX6 deficiency causes opacity, and yeah, I laughed when I saw this. It's like watching A Beautiful Mind. I mean, this isn't a summary diagram. This is a conspiracy theory. It reminds, although this meme wasn't around at the time, it reminded me of this. But what he's trying to show is that the PAX6 deficiency leads to oxidative stress, which leads to a chronic wound state in the PAX6 mutant corneas, which leads to dedifferentiation, loss of epithelial transparency, and opacity of the corneas. And that what we are hoping, our current work, which if I'm invited to speak again in the future, I'll be able to describe to you. Our current work is trying to push this diagram backwards, to start with the opacity, to reduce the oxidative stress, to calm down the mutant eyes, and restore the effects of the PAX6 deficiency such that the eye can survive. And the goal of our work is to create eye drop therapies such that when a baby is born with aniridia, it could start taking pharmacological eye drops that will prevent the corneal opacity from ever occurring, and therefore could potentially prevent the baby from ever going blind. That's all I'm going to say today. So it's been a little bit longer than I expected, but I did mess up the start, I guess. The story about what I wanted to say was that it's been now 32 years since the identification of PAX6 as the aniridia gene. And even now, large areas of the genotype-phenotype relationship are still unsolved. That's good for the young people out there, because there'll be work to do for the next 25 years in researching this and other genetic conditions. But what we have finally identified is that aniridic associated keratopathy is caused by a combination of oxidative stress in the cornea, and it's not primarily a limbal stem cell deficiency, although there is a minor deficiency of the stem cells which prevents them from responding properly to the chronic wound state in the cornea. Mutant limbal stem cells are slow to respond to wounding, but the good news is that they can do it, and if we give them a helping hand, they may be able to do it well enough in order to maintain the cornea during normal life. On the right here, this is my lab. They're all very shy. They didn't want their pictures. But I mean, the people, so Kaya and Eileen down in the bottom left here are the two who are doing the therapeutics on PAX6 mutant eyes to try and restore vision. And Jakob hiding at the back there is currently working on limbal stem cell activity. And Ahlam Alamri was the person who did the wound healing experiments on PAX6 mutant corneas. Everybody else, when they talk at conferences, shows a picture of how beautiful the place is that they came from. I unfortunately can't do that because I'm from Aberdeen. This grey pancake here, the so-called granite city, all the houses are built of granite, and that's kind of what it looks like. But it is, sometimes the sun does shine and sometimes it is a nice place to work. So thank you for being patient with me. That's all I've got to say. And if anybody's got any questions or any comments on what I've said, I'll be very happy to answer them. I'm going to stop sharing my screen now. Okay. Mm-hmm. I'm very happy to answer any questions, yes. Anybody there? All right, okay. Okay, so the question, what is the white ring-like structure seen around the cornea as we age? I don't know exactly the answer to this question. The limbal stem cells are a long-lived cell, and it's in the interest of the eye to prevent them from picking up mutations. So, which is difficult if you are an ocular surface cell, because you're exposed to light, including ultraviolet light, all the time, and potentially other environmental pathogens, mutagens. And so the stem cells have to be protected some way from mutation. And in humans, the way there's two mechanisms that do that. Well, three, but first of all, the limbal niche is pigmented, so that the kind of grey color of the limbal ring is pigment, which helps to protect the stem cells from ambient light and therefore reduces the amount of mutation. But also they're hidden away in specialized structures called the palisades of Vogt, which are little wrinkly projections of epithelium where the stem cells hide away at the bottom. And as we get older, these tend to get thicker and more wrinkly. It's a very slow process, but it does happen. And I think that thickening of the actual limbal niche, together with the opacity and the pigmentation of it, which protects stem cells from light, I think on the whole, that can be responsible for making the limbal ring more obvious as we get older. When one eye is injured, why do we also have activation of stem cells in another eye? That's the essay question, isn't it? I mean, who knows? What we think. Well, there's two answers here: mechanistic and evolutionary. If in the wild, how does this evolve? And acute corneal injury will usually happen in the wild in mammals if they're living in an abrasive environment, sandy, woody, a high matrix grass environment where one eye is subject to abrasion. And there's a good chance that if one eye has been subject to abrasion, the other eye has been subject to abrasion too. So evolutionarily, I can see it might be possible to evolve a scenario whereby the stem cells in one eye activate the stem cells in the other eye, because in the wild, it's not like in the lab with the surgery. In the wild, it's likely that if one eye is being injured, the other eye is being injured too. So I can see why it kind of makes sense. Mechanistically, how it happens, I don't know. When the eye is injured, the cornea, it releases growth factors such as HGF, EGF, KGF, and a number of cytokines that presumably get into the blood system. And maybe the activation of cytokine release from an injured eye travels systemically and reaches the other eye and activates stem cells in that eye too. I'm really only guessing at this. We never did the experiment to test this. And one of the interesting things I think would to do, if we'd thought of it, would have been to look at stem cells in other tissues as well. Because if the injured cornea is releasing growth factors and cytokines that activate stem cell activity, maybe the activated stem cells in the skin and the liver and and the gut and everywhere else, and we just didn't look at that. And I think it would be worth. It would certainly answer the question about whether systemic release of growth factors that is causing stem cell activity, if we can show that other stem cells within the circulation have been activated. Ooh, here's another excellent question. Does the stem cells lose efficiency as we age? Yeah, okay, so I didn't show you this. My first the earlier PhD student, Nada Saga, who looked at stem cell activity in wild-type mouse, she did exactly the same experiments, but on old mice. And by old mouse, I mean mice that are over a year old. Remember, most mice in the wild don't live to be a year old. In the lab, 12 months is well past reproductive age, and most mice will be dead by 18 months of age. They can live longer. So Nada did experiments on geriatric mice, and she found that yes, their stem cells in geriatric mice were showed much lower levels of activation in response to wounding compared to young mice. So if we extrapolate that to people, then we would guess that yes, stem cells do lose efficiency as we age, and that in old people with injured eyes, we would expect lesser of a wound healing response, which fits with our sort of general dogma that old people heal less efficiently than young people. Sorry. That said, there is no our corneas don't fall apart just because we get old. Most old people have corneas that are just as transparent as young people. Just because you get old, your cornea doesn't necessarily go opaque. Lots of old people have perfectly good vision. So if there is a reduction in stem cell efficiency with age, it's not enough to cause degeneration of the cornea. But it may be enough to reduce the wound healing response to injury in older people. Well, thank you for being patient with me, and it's been a real privilege to be able to speak to you today. Oh, that's okay. Ooh, that's very nice. Thank you. That's really lovely. Thank you. That's brilliant. Thank you very much. It's been a privilege to speak to you today. Thank you. Okay. I will turn off my mic and camera, and I hope to see you. I hope to see you again soon. Thank you. Mhm.
Thank you, ma'am. Very good evening to all present. It's my immense pleasure to introduce our guest speaker, Dr. Akshay Murugesh, Faculty of Medicine Health, Department of Biotech, Tampere University, Finland. She had completed her Doctor of Philosophy in Marine Biotechnology from Annamalai University in the year of 2015 and a Master of Philosophy in Biotechnology from Bharatdasan University in the year of 2007 and a Master in Zoology and a Bachelor in Zoology in the year of 2006 and 4 from Dock College, Lady Dock College. And she is currently working as a PDM fellow in molecular signaling lab with Tampere University from Finland. And she had experience as an assistant professor and a coordinator for research and development cell and instrument purchase and maintenance. And also she had experience in research associates and project trainee. And she is having many funding projects like DST SERC, DBT, and UGC, and also ICMR. There are many publications in the national and international peer-reviewed journals. She has supervised many UG, PG, and PhD students, scholars. She has given many invited lectures and served as an examiner for UG, PG, and MPhil thesis evaluation. Once again, thank you all for giving this opportunity.
Thank you, Ms. Kirtana. Okay. So, whether my slides are visible to everyone? Yeah. So, my presentation is on often purinergic receptor signaling, GPR17, a modulator for treating glioblastoma. So my lab is focusing research on glioblastoma, and I'll give you a gist of intro of what is glioblastoma. Then I'll focus on the research, various research that's happening on glioblastoma. So this is a very basic intro of what is the brain tumor facts worldwide, as well especially in Finland. So globally, when you observe, or globally or regionally, or as well in national level, the burden of brain tumor, that is this, the first graph, the statistics, which is between 1990 to 2016, it says that there is a gradual increase in the burden of brain tumor, and mostly, you know, like the male is very much prone to the tumor than the female, and this might be the fact that of the lifestyle habit. And when you see the next statistics, it says like how much of new cases that comes in every year. Like this is the association between the incidence of the brain tumor and mortality of the brain cancer. You know, like we call it as HDI, which is Human Development Index. So mostly, you know, like Asians are very much prone to this, which is around 52.6%, and second comes the European countries. Even the Finland comes in this Europe, which is around 22%, and you can see other countries with the less, that is 10 and less than 10%. So you can't see much difference between the new case every year or new case and the death penalty, because the main challenge of this disease is once it has come, or when an individual is getting this disease, it is not curable. So it's very, very challenging disease, and their lifespan is just 1 to 1.5 years. So intense research is going on for the therapy development. So this is again a statistics which is mostly on the Finnish Cancer Registry of 2020, and this compares the number of new cases and year-wise statistics of men and women. And as well, from the graph, you can see the different types of cancers like breast, I mean, like for women, the breast, the top leading cancer, and for the men is the prostate. In Finland, the prostate is the top leading cancer, and the other cancers like even the second leads the bladder and urinary tract for the men, and corpus uteri for the women. So likewise, like the statistics, this is exactly the statistics of Finnish, and again, I focus most on the statistics of the Finland because we mostly concentrate on this particular research of Finland, but the development of therapy is totally for the global use. And this is 5-year data of the brain cancer diagnosis, and almost new. The red color bar says that around 37.6% of people in Finland, they've been diagnosed with, adult. It's between the year of 2000 to 2014, they have been diagnosed with a brain cancer. You can see other countries too, where there's almost like about 20% of the brain cancer incidence was observed. So coming to the basic facts about, the basic classifications of the brain tumor. Basically, the brain tumor is classified as the primary brain tumor and the metastatic brain tumor. Our group focus mainly on the primary brain tumor, and the primary brain tumor is nothing but the tumor which actually been localized, they develop exactly in the brain, different any part of the brain. And whereas the metastatic brain tumor is like, it can be in any part of the body, but the cancer cells can move from other parts of the body to the brain, and they can cause all kind of symptoms related to the brain tumors. And coming to the further classifications of the brain tumor, primary, especially the primary brain tumor, which actually we are focusing on. This primary brain tumor is the one where the cells, we call it as glial cells or the gliomas. Those are the cells which becomes cancerous, which are very much similar to glial cells. Glial cells are nothing but, those cells which are present around the nerve cells. So these gliomas, they are very much similar to glial cells, and they develop into tumor. They differentiate very fast, and they develop into tumor. So based on the nature of its differentiation, it's been further classified, the severity also, it's been classified into 4 different types: the low-grade astrocytoma, malignant astrocytoma, glioblastoma, and oligodendroglioma. So of which the glioblastoma, which I have given here as a grade 4, this is a very dangerous irreversible type of cancer, which we actually focus on. Our research actually focus on this particular type of glioblastoma cancer. So according to WHO, those individuals who have this grade 4 type of cancer, which is a glioblastoma multiforme, we call it as GBM. So this type of cancer is very dangerous, and their lifespan is very, very less. And as I said, it's a very simple definition of the glioblastoma. It's a collection of abnormal cells, especially from the brain, or it can also arise from the spine canal. And as I said, the incidents are increasing every year, and their death incidence is like incidence of case of diagnosis is 308, and its death case could be like 1 or 2. And their mean survival rate it's around 12 to 18 months, right? And coming to the symptoms, the symptoms, as I say, it is in the brain. The brain is the master of our body, which controls all kind of activities, thinking process, and everything, whatever we perform, right? So the symptoms are, they will have an unusual headache, often seizures, memory loss. They'll have some kind of personality disorders, will not be very, very normal to the normal. They won't behave very normal as with respect to the normal individual. And they can't process any kind of information perfectly. They'll have a blurred vision problem too, and change in motor control. So almost all the functions, wherever the tumor is localized, and accordingly the symptoms might vary, the localization, based on the localization. So almost all the functions, they deteriorate based on the stage and the severity of the cancer. And how this disease is diagnosed is, we say like gold standard method is MRI scanning is a gold standard method. And in an emergency case, they go for CT scanning. Then, as usual, of biopsy, like removal of samples from the place of tumor formation, and it will be sent for analysis of the biomarkers. Biomarkers which says that it is cancerous cells. And sometimes, body fluids also is being used for the diagnosis. So, coming to the treatment strategies, very usual treatment strategy for the cancer: surgery, then chemotherapy, radiotherapy. And this could be of a single type of therapy, or it can be a combination type of therapy based on, as I said, the severity of the disease. And what are the other factors which really lead to the cancer cell formation? I mean, like the features I can say are as the hallmark of this particular type of cancer. This cancer, the tissue invasion from one place to another place is very, very high. So they are highly metastatic. For glioblastoma is highly metastatic. That's why their median life or the mean survival rate is very, very less. And their microenvironment will be inflamed, and nothing like in the growth inhibitors, any kind of growth inhibitors will become insensitive. And other factors like sustained agenogenesis or evasion of apoptosis. So these are the hallmarks of this particular type of cancer. So as I said, we were discussing the basics of this, the challenges that the individual or the patient really face for the treatment. Why the median survival rate is very less is because of all these important challenges. And the first important challenge is the blood-brain barrier. I hope like everybody is familiar with this term, blood-brain barrier, because your brain is so protective, it won't allow anything to enter in. So it keeps the organ very safe as a barrier. So when a drug is given, again, this happens, this functions in another way, like this barrier will not allow the drug to enter into the brain. So, the localization of the tumor, it cannot be targeted by the drug what is given. So the treatment will not be completed. So that is a biggest challenge with respect to glioblastoma. And coming to the tumor heterogeneity, as I said, again, the environment might be based on the localization, the severity changes. The cells or the cancer cells would be of different types of heterogeneous groups of cells would be there. And when you give a drug, one type of cells will respond and another type of cells will not respond to the drug that's given. So targeting all type of cells, a heterogeneous group of cells, is very, very important, even though the drug crossed the blood-brain barrier. And the prognosis is very limited. And we have a lot of communications with the local environment. Like as I said, the communication signaling process would be very, very high. So the process of prognosis is limited, and it hampers the diagnosis process also, and the disease progression is very high. The proliferation becomes very, very high, and progression of the disease is very fast. And all these ultimately lead sometimes, it depends on the individual's genome thing. Personalized factors also plays an important role here. Some people will show up resistant to a particular drug in a very limited period of time, and some of them will so like in a 1 year or 1.5 years. So the drug also will become, most of the time it becomes resistant, and recurrence of the disease is most. So these are the challenging factors. Now coming to the research in our team is, we actually focus on all type of cancer, and today I'm going to talk about the research focusing on the glioblastoma. So based on all these research, the research on the different type of cancer cells, we specialize our research on glioblastoma, and the compounds that we have taken for our study is the phenolic compounds. The phenols are, we know that it is a very good agent, chemotherapeutic agent to treat, it can induce apoptosis, it can cause stoppage of cell division. So a lot of good features are there. So it is considered to be a potential compound to inhibit the growth, further proliferation of the cancer cells. So we have I have given here three different compounds, which is it's very briefly mentioned over here, and if you are interested to know what it is, you can just visit the website of Molecular Signaling Lab of Tampere University to have the expansion of these compounds. And these are the references of my colleague who actually worked on this. And these are the three important phenolic compounds we have intensively studied, and based on the R moiety, that is the group that is present in the R, which is either methyl or O-methyl, the IC50 value, or you can say its toxicity to a particular cancer cell varies. So overall study of the basic, generally we start with, I'll tell you what is the workflow that we have done, but generally when we do in vitro analysis, the first thing and the foremost thing we did is selection of compound, and then we will go for how these compounds really have impact or really induce apoptosis or cell death in that particular cell line, and the IC50 value. That's a very important point to understand. So out of this, we come for a conclusion that THTMP, this is a particular compound, is found to be more effective with the least IC50 value. So maybe I can explain you with respect to another. So coming to the next concept, which is on GPCR, which is a G protein coupled receptor. This is a receptor signaling pathway, and it's a very good target for most of the multiple diseases, and especially multiple sclerosis or traumatic brain injury. So the right side you can see the expression of RNA and the expression of the protein of this particular GPCR receptor. You can say as in specific, that is GPR17. GPCR is the biggest family, of which the GPR17 is one, comes under the GPCR. So we also want to focus on how this GPR17 influences whether it overcomes the challenges what we have seen earlier. So we don't want to either stick on to only the compounds that really causes cell death. We also want to go with combining the other factors, like here we comes with GPR17. So as I said, the right side you can see the RNA expression as well the protein expression of the GPR17, of which the brain seems to be the toppest expression pattern. And in the left, I have given a simplest schematic representation of how the GPR17 signaling pathway is activated. So the toppest is without any ligand binding, and the bottom one, which says that when a ligand binds, you can see a phosphorylation happening, which induces a lot of expression of other genes at the bottom that ultimately leads to the cell differentiation, right? So this is how the signaling happens with respect to GPR17, and we also want to understand the combination of THTMP, which is the the topest or the potential compound, and we also want to combine the ligand, which actually has the ability to bind with the GPR17 receptor. So combining both of these together as a combinatorial, what would be the impact on the cancer cell lines, especially on the GBM cell lines. So our hypothesis is surgery or radiation or chemotherapy, generally when all these protocols being followed, temozolomide, I just want to talk about temozolomide over here, which is the chemotherapy, common chemotherapy used for this kind of cancer. This temozolomide, in general, it also has impact on the normal cells, and so it has a very, even though it kills brain cells, cancer cells, it also kills the normal cells. So we're looking for the better options, always going for a better option which doesn't have any impact on the normal cells. So now the right side, you can see the right side of the slide where the phenolic compound and the combination of the GPR17 receptor, we also worked on a lot of compounds which actually targets the GPR17, and the top leading agonist, we call it as agonist, which actually gives a positive signaling processes upon binding with GPR17, and T0 is a tag of name or symbol we have given for the compound, which actually which is actually a potential ligand binding with the GPR17 receptor. So we propose, we plan to give in a combination of these two compounds, and we just want to see what would happen with respect to the other molecular processes in the cancer cells, especially in the glioblastoma cells. So this is the workflow that is done to complete this particular hypothesis. And workflow, it starts with the docking simulation, which fits best those compounds were taken, and immunoblotting, other secondary messenger analysis, and its cytotoxicity, and a lot of in vitro assays, which is a standard assays essential in order to analyze the efficacy of a particular compound, which is like migration. It should not allow the cells to migrate. When you add a compound, the brain cells, or sorry, the cancer cells should not migrate, which is directly correlated with the concept that it stops the migration and invasion, and as well the colony forming, which is the ability to form the tumor, they can attach with the nearby cancer cells and develop into a tumor. So all those assays, and along with apoptosis, ability of your drug inducing the cell death, that was also been done. And we also want to understand how it really influences cell cycle pathways, which is the basic cell cycle pathway, where it actually influences either in S phase or M phase or in G phase, where it actually plays a role in stopping the cell cycle, and other free radical analysis like ROS, MMP, caspase. So these are the in vitro analysis. And apart from the in vitro analysis, the next stage of analysis like RNA sequencing, protein profiling. So these two stages of analysis will tell what are the genes which really being influenced by the drug, and what are the genes which plays a vital role in particular cancer and in different stages of cancer. All those things can be analyzed by analyzing the RNA sequencing and proteomic profiling. And finally, every study involves a xenograft model. It can be authenticated when you do something, even though you do a lot of experiments in cells, it has to be authenticated only once when you do it in the animal model. Then after successful treatment or successful experimentation or results in the animal model, it could be taken for the preclinical analysis. So this is what we have done. This is the workflow. So coming to, I am not going to show you the pictures of the graphs or the animal models. I'm going to just explain the 4 important work which is related to the concept which I have explained previously, So this is the first objective. As I said, the top 3 compounds we have talked, we have analyzed those top 3 alkyl amino phenol on the GBM cells, and we actually analyzed all these factors which is given at, as I said, the hallmarks of cancer. Those properties were given at this side. So what was identified is, like, as I said, the top compound as the THTMP. THTMP has the higher cytotoxicity, which is like it can kill the cancer cells more than the normal chemotherapeutic agents, that is temozolomide. And it also induces the DNA damage by P53 pathway. The pathway that it takes to damage the DNA is P53. And as I said, we also have done the cell cycle analysis. So it was able to stop the process of cell cycle in the stage of growth and synthesis phase. So it cannot enter to the next stage. And it has the ability to decrease the cyclin-dependent kinase 1 or cyclin A2E1. These are the genes which was analyzed through RNA sequence analysis, and it has a good, these are the genes which whose expression was very higher in the cancer cells, brain cancer cells. And this compound, which is THTMP, was able to reduce the expression of this genes, and it was also able to induce apoptosis, which is the cell death, and that was actually regulated. We actually correlated the genes or the proteins which induces some kind of processes. Okay, when it arrests the cell cycle, those are the cyclin dependent, cyclin A2, cyclin E1, E2, or those genes which actually whose role is being decreased, and because of which the cell cycle arrest happens at that particular stage. And as well, it induces apoptosis, and this induction of apoptosis is purely by the regulations of the genes which I have given at the top, at the bottom, which is like BCL2G or ROS, which is free radical dependent, responsible gene, and some of the apoptotic genes, which was explained in the paper which I have given at the top, right? If you want to know or understand in an elaborate way with the diagrams, you can access that. It's a gist of those results, what we have done it for those 3 alkyl amino phenols. And the second objective is we characterized the anti-cancer effect of the top leading compound THTMP on the glioblastoma cancer stem cells. As previously Professor Martin was also talking about the stem cells, and these stem cells, they have a very vital role in the prognosis. And we were actually, I was discussing about the heterogeneity of the tumor cells. So what we have identified is, like, as I said, the population of the stem cells would also be different, and in a particular tumor and microenvironment, and those would be, like, we call it as GRC, which is like glioblastoma stem cells, which is the cancer cells, and non-glioblastoma stem cells, which is the non-cancer cells. So you will have a mix of population of glial cells, cancer cells, and non-cancer stem cells will be there. So this THTMP, the drug top leading compound, what we have identified. So what initially we did is we have separated those 2 cells from those microenvironment, and those 2 cells were individually treated with this particular compound THTMP. And it was also compared with the normal drug. It's very important that the drug, or it's very important that any kind of compound, you can say it is potential when it works better than the existing drug, right? So we also want to compare with the temozolomide variants. What is here given as a variance is, we tried developing resistance ability for the cancer cells, brain cancer cells, to this particular temozolomide. And we also created sensitivity of cancer cells, sensitivity of the cells to this particular temozolomide. Then the resistant cells and the sensitive cells, those two cells were treated with the THTMP. And even if it is sensitive or if it is resistant, this THTMP, which we proposed as a very top leading compound, it is able to sensitize, which means it is able to target any type of cells. This particular feature is mostly related with the fact of what we see in the previous slide. Cells becomes resistant to the drugs. Okay, so when it becomes resistant, as I said, the treatment becomes failure. But when we tried with different sensitivity of the cells we created and we treated, it was very, very effective. And this compound was able to arrest the cell cycle at G1S as well in G2M, both the stages. And it is specific for different population. G1S for GSC, G2M for NSCC cells. So even for different populations, it was able to, according to the nature of the cells, it was able to stop. And it is able to induce apoptosis, cell death. We were able to identify that this compound was able to target EGFR as well the cancer stem cell signaling pathways. Also, the downstream signaling genes are there, and a lot of these genes are influenced, which are influenced in a positive manner by this particular compound. And so the induction of apoptosis was very, very successful. So whatever I have explained, I have correlated the features, like what is actually it is inhibiting. I have given it as a cross. And coming to the next combination effect, we have studied separately, and we want to, as I said, we want to combine these two compounds, and we want to check the combination combinatorial effect, whether it is working better than the individual THTMP or it is working good. And from the results, the combination, always a combination works better than the individual. Yes, of course, it showed a strong inhibitory effect with the least IC50 value, and it reduces all the migration, invasion, or colony formation, which are the special characteristic features of any type of cancer cells. And it arrests the cell cycle at S phase of the cell cycle. And other intracellular messengers like calcium and MMP, they were all increased, which actually leads to the process of the apoptosis and other molecular processes. And we were able to identify the pathway, what is the pathway it really takes up for induction of apoptosis. There is always an intrinsic and extrinsic apoptotic pathway. The pathway it takes is intrinsic pathway to kill the cells. And this we have taken it to the xenograft model. Xenograft GBM model was developed, and it was injected with this combination of the drugs, which showed a very equivalent, promising effect in reducing the tumor volume. As well, it showed increased survival rate of the animal when we given this combination of the drug. So when you see combination, too many circles, which is like too many potential features, when you do it in a combination, could be achieved. And the final the work with respect to this particular objective is We also want to identify how the expression pattern also, like when you add something, it does something, okay, and how actually it influence the expression. We could see that GPR17 expression is very high in the brain. From the previous slide I have said, so how this expression, endogenous expression, also helps in the survival as well in the treatment of GBM. And we first confirmed, yes, of course, there is an endogenous expression of GPR17, and we also found out that it showed higher cytotoxicity effect than the temozolomide existing compound. And this mediation of GPR17 activation, it happens through a lot of genes. I have given here as P13K act and MAPK, STAT, and NF-kappa B. So these are the major genes which is involved in the activation of the GPR17, and it was able to induce apoptosis and arrest cell cycle at G1. And very interesting factors, like it was able to even cross the blood-brain barrier, and we have gone for an additional examination here is like we have got the patient-derived GBM cells, and we were able to analyze the same data in the patient-derived cell lines as well in the xenograft model, which showed a promising results. So the expression also helps in giving the better results. So coming to the summary, from the previous slides, I've just made a gist of like a combination of these compounds was able to give or it increases the anti-tumor ability of this particular compound. It's combination of the THTMP and GT0 compound. So the significance of our work is like we know that current drug, it doesn't shows efficacy on the heterogeneous group of population of cells, or it develops resistance to the drug, or recurrence is happening. And the solution that was like at present solution that is happening is like people are managing. I could say managing is like multiple targets. They were managing with temozolomide because it was able to target the multiple tumorigenic features and inhibits the multiple signaling pathway. But the exact, what we have identified through our research is like we were able to suggest that alkylamino might, along with GPR17, as a new target for GBM treatment, and the combination must be a promising candidate for GBM drug development. Thank you. So. Yes, please. It's not like that. Actually, this drug was we did in all types of cancer, as I have given in the initial schematic representation. We worked with the breast cells, we worked with the colon cells, we worked with even liver cells, H329 cells. But all the cells, it had its cytotoxic impact. But since we want to specialize our research in glioblastoma, we did very extensive work in this, and our collaborators are working on the other cancer cells also, which is giving very similar data. So it's not so specific to one type of cancer. This GPR17 is actually, this target is very a good target, and it's working for all type of cancer. So it's not as specific for GBM alone. Yeah, yeah. So the connection was there when I answered the question, right? Yeah. Yes. Yeah, yeah, yeah. So we were successful with the xenograft models, and we were actually trying to patent the compound, and the group is actually trying to patent the compound, and it's patented actually in the Tampere University, but making it commercial is, it involves a lot of process. But before that, what we are planning is we would like to take it up to preclinical analysis, to human samples preclinical analysis. So it involves a lot of medical clearance, and especially in a country like Finland, it involves a lot of medical clearance. The group is working on it. So hope it has to clear, and it works better. We hope that. Yes. Welcome. Yes. Yes. Thank you.